so good morning, everybody. Good afternoon, maybe for some of you. And it's a real pleasure on behalf of the Columbia and Berkeley Superfund Research Programs to welcome all of you to this mini-symposium today on metals epigenetics. So for those of you who don't know me, I am Ana Navas-Atien. I am a professor of environmental health sciences at Columbia University. And I also have the honor and the challenge of serving as the Columbia Superfund Research Program Director. And so the mission, before we start, I would just like to talk a little bit about what is the mission of the Superfund Research Program and how the mission is to provide scientific solutions to protect populations from hazardous substances. So to meet that mission, scientists, we are generating a lot of data. I think it's been made very clear to all of us during this COVID pandemic, and this applies also for our very chronic environmental health problems, that is not only generating data, which is of course very important, but also it's very important to extract valuable information from the data that we generate, so that we can actually translate those data into solutions for our problems. And this is a scientific and also an ethical responsibility towards our participants who donated their biospecimens, to ours and the communities, the funders, of course, and the society at large. So this is something that needs collective effort. There is not one single scientist who could do it alone. And extracting valuable information from our data really need all of us together. So this is the reason why, when we were planning this symposium with Andres Cárdenas from the Berkeley Superfund Research Program and a faculty at Berkeley, and I will let him introduce himself in a little bit, and myself, we thought it would be really amazing if so many of us who are working on metal epigenetics, we could come together and start discussing what we are doing, what are we learning, what are the challenges, how can we best extract the hidden information that is in DNA methylation or other forms of epigenetics data. How can we learn which studies have comparable information? How can we learn how to pull data better, how to increase power, evaluate the robustness of the findings, evaluate generalizability and the sources of heterogeneity? So that's the reason why we thought, let's come together and have this discussion. As the internet allows to disseminate these ideas and bring people together, we realized that from our very small group of people that we started this, our stellar presenters and Andres, myself, and Nancy Loiacono, who has been fundamental in making all of us being here today, we realized that there was a lot of interest, and I can see now we have more than 100 people who have joined. So thank you so much, all of you, and we hope we can have please introduce yourselves through the chat. Let us know who you are, for those of you who just joined. And let's have a wonderful, exciting discussion, learning about which metals have been studied, which different tissues have been studied, which types of epigenetic modifications have been studied, and how can we do this better together. And so Andres, please, maybe if you would like to introduce yourself and also talk a little bit about what we are going to be doing and introduce the speakers, and thank you so much, everybody.
Of course.
Andres,
yeah. Good morning, everyone. Well, I'll say good morning because it's 8 a.m. here, but thank you so much for joining us. I'm Andres Cárdenas. I'm an assistant professor of environmental health sciences and computational biology at UC Berkeley. And as Dr. Ana Navas mentioned, we're very interested in forming collaborations. We're going through this exercise with both of our superfunds right now to try to integrate data and really collaborate at the data level where we can normalize and integrate the data so we can make better inferences at the end of the day. So we would like your input at the end of the day. Today we'll have a little discussion about how we can collaborate and move the field forward, so we're not working in silos. So as Ana mentioned, this is the main objective of today, and we are fortunate to have an amazing lineup of speakers. To start with, I would like to introduce Dr. Carmen Marcet, who is our first speaker. Dr. Marcet is a professor of environmental health at the Rollins School of Public Health at Emory University, where he also serves as the Associate Dean for Research at the Rollins School of Public Health. And I would like to mention that I actually met Dr. Marcet or Carmen as a trainee in the Superfund. I was a trainee at the Oregon State Superfund, and I was very fortunate to do a Casey Donnelly externship where you go to another lab to work with someone. And I was very fortunate that Carmen took me in, and I spent a little bit of time on his lab when he was at Dartmouth. And he's a fantastic collaborator and scientist, and many of us have been following his really great work on epigenetics. And he will talk about using placenta to understand the developmental impacts of metals. So Carmen, thanks so much for being the first speaker, and take it away. You have about 10 minutes. I also want to encourage the participants to post questions on the chat. I'll monitor the questions, so Carmen or the presenters don't have to worry about following the questions. At the end of the presentation, we'll try to summarize the questions for the presenters. So thanks so much, and Carmen, please go ahead.
Thank you very much. I very much appreciate and Andres for bringing this together. I think this is a great opportunity to hear from really great speakers, so thank you. So I'll tell you today a little bit about the work that we've been doing in the placenta to understand the developmental impacts of metals. And we're really interested in the placenta because it's the first complex organ to form. It comes from this outer layer of cells of the blastocyst, the trophectoderm, that invades the maternal decidua and eventually forms this branching structure that allows for the exchange of nutrients between mother and infant. But the placenta does a lot more than just really the exchange of nutrients. It is this transport organ, so water and waste and gases have to move between mother and developing fetus. But it also is a very active organ in the production of hormones and growth factors, different types of monoamines with neurological properties. It's a metabolic organ. It provides immune tolerance, and then it performs a number of sequestration and barrier functions. And in some cases, that is around metals. So our lab has, for the past almost 15 years now, been trying to understand more about how this placenta works and how different exposures from the maternal environment might be impacting the functions of the placenta. And we use functional genomic methods to try to understand better what the impacts of different types of environmental exposures are on the placenta and its operation, and then how those might be impacting children moving forward. And so, we've interrogated everything from DNA methylation in the placenta we've done a lot of work on transcriptome with Maya Dyson-Roth, who's now at Columbia. And we're now doing a lot of work today on microRNA expression. And so that's what I'll talk a little bit about, the work that we're doing with microRNAs. This is work done by a number of people in my lab, so I have to give them credit. I just get to talk about it. This is a great collaboration with Devin Kessler at the University of Kansas Medical Center, and then some really talented postdocs and students in the lab, Elizabeth Kennedy and Fuying Tian, who are postdocs, and then Jesse Tarani, who's a new PhD student in the lab. And they're really the people working on a lot of these analyses. So we've known for quite some time that microRNA, these are those small non-coding RNA that can affect the expression of genes and the translation of genes into protein, that they are responsive to the environment. This is some work I did as a postdoc in cell culture, demonstrating that a number of different stressors can impact the expression of microRNAs, and this was in cell lines at the time. But we've moved on to then examine a number of these different stressors in human populations, and I know Andrea has also published a number of papers looking at how environmental factors impact microRNA expression. MicroRNA are actually really unique players in the placenta, and they're really very closely involved in placental development and differentiation. They play major roles in trophectoderm development and in the cytotrophoblast to syncytiotrophoblastic transition. So that's when these cells start to fuse together to allow for better exchange of nutrients. They also play roles in invasion and migration, in angiogenesis, and in growth. And in fact, there are two primate-specific classes of microRNA that are specific to the placenta. So these really evolved with placentation in primates, and are specifically expressed in placental cells. So this is the chromosome 19 and the chromosome 14 clusters. And there's been a number of studies, a lot of this coming out of Yoel Sadowski's group, looking at these clusters and what they do. And in many cases, they've actually been shown to be expressed in the placenta and then transported in exosomes into maternal circulation, where they may be affecting maternal immune function. So there could be a lot of different functions for these microRNA in placenta, both in the function of the placenta itself, as well as a means of communication between fetus and mom. So for this work, we've done placenta whole microRNA sequencing using small RNA-seq. In this, we're using two different populations so that we can be able to reproduce our work. So we use samples from my Rhode Island Child Health Study cohort, which is a hospital-based study from Rhode Island, and as well as the New Hampshire Birth Cohort Study, which is in collaboration with Margaret Carragus at Dartmouth, where we've been studying placentas from her population of women exposed to well water during pregnancy and potentially having contaminants with various trace metals. Using these sequencing approaches, we identified about 600, or almost 700 microRNA that were expressed in both of those populations in the placenta, and after a number of quality control steps. So some of our first studies, this is the work by Liz Kennedy, and this is now, we have a preprint of this paper online, and it's under review right now, looking at how these microRNA are associated with birth weight. So one of the key questions we want to know is, can we actually identify specific associations with some important outcomes with these placental microRNA? And so looking at that, we identified a number of microRNA that we could reproduce across these two cohorts associated with birth weight, and the figure to the furthest right shows the meta-analysis between the two cohorts, and this MIR532 is what I'm highlighting as one of the strongest effects that we saw between the two cohorts associated with birth weight. We're then able to go on, because we have the whole transcriptome data on these samples, we use some bioinformatic approaches to look at what the targets of this microRNA are. Then we can integrate in our whole transcriptome data and actually bring in gene expression to identify if there's actually physical changes to the transcripts themselves. And what we found is that leptin and adiponectin seem to be the major targets of these two microRNA. And that sort of makes sense when we're talking about something like birth weight and growth, that we may be affecting this leptin and adiponectin pathway. So that associates with birth weight. We also want to now go on to look at what some of the exposures are that might be driving variation in different microRNA in the placenta. And we've started some work on placental cadmium. This is the work that Jesse is working on right now as part of his thesis, looking at how placental cadmium associates with these microRNA. And we identified a number of these microRNA associated with placental cadmium. And placental cadmium is interesting because it's sort of known to play some of its role in fetal toxicity through the placenta. It can be accumulated within the placenta, and actually does have some toxicity to placental cells. And so it's an interesting toxicant to look at for placental effects. And so what we identified were some of these microRNA, including these 10B microRNA here, which have previously been shown to be downregulated in preeclampsia. So we're identifying that microRNA, associated with this very significant health effect in women, are also downregulated by placental cadmium exposures. And some of these other microRNA have been shown to be affected by things like nutrient stress. And so it does demonstrate that there does seem to be some toxic effects, potentially of cadmium through impacts on microRNA. Now, on the other side, we're also interested in how some nutrient metals or antioxidant type metals might be impacting microRNA as potentially protective factors. And so we looked at selenium, and this is the work that Fu Ying is working on. And what she identified was microRNA, specifically this 216 and 217 microRNA, as being downregulated. And we can see on the right, it's, selenium is one of these metals that may have sort of non-linear properties when it's affecting health. And so we also wanted to look at potentially non-linear effects on microRNA expression with selenium. And what we can see is that there's generally a downregulation of these two microRNA associated with increasing levels of selenium in the placenta. And this is interesting because these microRNA have previously been shown to be increased in related to oxidative stress in a number of different cancer cell lines. And so it looks like here that selenium may be playing that sort of antioxidant role in reducing the expression of these microRNA that are generally increased with oxidative stress responses. So we're kind of really excited about this potential for microRNA in the placenta to play a major role. We think that it could be playing a key role in regulating some of the toxic effects of metals, and then regulating the downstream outcomes that may be related to those metals on children's health, both at birth and then in the long term. And in general, we kind of see this, the placenta itself as an opportunity to really integrate, genetics and epigenetics to try to better understand all of the effects of the exposome and how this is impacting long-term child health outcomes. And I will stop there and just thank my lab and my fantastic collaborators that really allow us to be able to do this kind of work. And I'm happy to take any questions.
Wonderful. Thank you, Carmen, so much for this great first talk. So everybody, please send your questions through the chat as we move on with the different presentation. Feel free to post your questions in the chat as the speakers go, and you see a slide that is particularly intriguing, or you have a particular question. I think that will be very good. So while people put some questions, Carmen, I have a question myself. So you showed some of the microRNAs that were related with the low birth weight or with birth weight. And then you showed some microRNAs that are related to the metals, to cadmium. Are you trying to look at those together? Is that, or maybe you did and I missed it? And like, how is this coming together? Or how is
eventually that's the whole plan, right?
do you have enough power to do it? Like,
yeah, I mean, the challenge is we probably don't have the power that we need to actually look at the full range of mediation for some of these effects. And particularly because, I mean, again, we are looking at also at populations that are, these are US populations. So we're not necessarily looking at extreme exposures to metals, but more what the general population might be seeing. And particularly something like cadmium, in fact, both New Hampshire and Rhode Island have very similar levels of exposure to those two metals, because it's probably not coming necessarily from their drinking water, but from some other sources. Mm-hmm. So yeah, I mean, ideally that would be the ultimate goal of a lot of this is, can we identify some of the exposures, and hopefully, we're kind of thinking about different exposures that we can be looking at for these birth weight related genes and how they might be impacting them. So.
Very good. And there are two questions here. One from, I don't know if you can see them in the chat, but one is from Susan Corrick. Are there alternatives to placental tissue that could be used to study the microRNAs? So like extracellular vesicles? Yeah,
definitely, Susan. Absolutely. In fact, I have some work with Carrie Brenton at USC where we're looking specifically at extracellular microRNA during pregnancy from mom to try to identify if there are changes that we can identify throughout pregnancy that might be related to exposures, and then be able to be more predictive, for example. So, that is the other challenge with even thinking about mediation. In this case, is that this is all very cross-sectional. So we don't really have some of the causal framework that we can really apply. So a little bit of it is hand waving.
Very good. And then a question from Jane Cleal. Which placental cell type are these microRNAs changes in? Have you looked in trophoblast culture experiments? Yeah, so we have not. So these are from full thickness sections of placenta. So it's really placental parenchyma. And so we have not teased out exactly which cells that we're looking at, but that could be a really important issue is, which cells are really being affected by this. And so it'd be interesting to look in some of the trophoblast experiments to better do that, so. Okay, very good. So thank you so much, Carmen, for your talk. And please stay here so that we can continue the discussion as we move toward our final comments and panel. And now I would like to introduce Dr. Bob Wright, who is a professor and the assistant chair of environmental medicine and public health, also a professor of pediatrics at the Icahn School of Medicine at Mount Sinai. And it's a tremendous honor for us to have Dr. Bob Wright here with us today. He it's impossible to summarize Bob's research in just a few sentences, because his work is so broad and so important for environmental health. And so we are really honored. I would just say that we are also, in addition to many of his numerous appointments, he also serves as the chair of our external advisory board for the Columbia Superfund Research Program. And that's a service that I am extremely grateful for, because his advice and his insight is always extremely important. So thank you, Bob, for joining us today. Do you have are you unmuted? We cannot hear.
Hear me now?
Perfect.
Okay.
We can hear you perfectly.
Okay. So thank you so much for that kind introduction. And I realize I only have 15 minutes, so I may talk a little fast. I wanted to talk about a topic that I think everybody has probably at one point measured or studied, but maybe hasn't thought really deeply about what they actually do. And I actually think they're really, really interested in. These are repetitive elements. And these are often defined as transposons and retrotransposons. And they're repetitive DNA sequences that actually make up more than half of all our DNA. And evolutionarily, they originated from viruses. So viruses use us to replicate, and in that process, their DNA got inserted into our DNA, and in some cases, it became replicated over and over again. So you can tell yourself that you're actually more than 50% virus, because more than half of your DNA actually came from viruses. And retrotransposons are actually the more common repetitive element, and they originated from retroviruses. And probably the most famous retrovirus is the HIV virus, and it's shown here. Infecting a cell and transcribing and expressing reverse transcriptase in an endonuclease so that it can replicate. And that's exactly how retrotransposons work in our bodies. There are two very common retrotransposons. One is called ALU. It's a type of sign, which stands for short interspersed nuclear element. About 10% of our genome is actually the same short sign, repetitive DNA sequence over and over again. And then there are lines, which are long interspersed nuclear elements. And the most common of the lines is called line 1. So these elements actually have very boring names, which is probably a function of how they were thought once to be just junk. But line 1 actually makes up 20% of our genome. So more than 50% of the genome in total are repetitive elements, and more than 1/3 of all DNA methylation actually occurs in repetitive elements. And we used to think of it as a way to measure sort of baseline or global DNA methylation. And actually my interest in this got started because I started to wonder, well, why would you bother to methylate something that was junk? And it turns out there are reasons why you want to methylate it. So it's often thought of as global DNA methylation. That's probably not a very good name because it's not really reflecting global methylation, and it really does have a function. So repetitive elements have a similar consensus sequence in each element. There are very common assays that are used to measure their methylation, but remember that there's hundreds of thousands or even millions of these. So what you're actually measuring is an average methylation in an average across a population of cells and across a population of multiple sites, and that there's tens of thousands of these. So it's an average of an average of an average when you're actually measuring line 1 methylation or ALU methylation, which means you're probably losing a lot of information because you're averaging it out so many times. So line 1 repetitive elements are particularly interesting because they can be transcribed and translated, and they actually code for proteins. They have very boring names: ORF1 and ORF2, open reading frame 1, open reading frame 2. But these proteins actually have endonuclease and reverse transcriptase properties, just like the HIV virus in its genome. So they can be expressed and they can be inserted across the genome. And this illustrates how that happens. So you have a line 1 sequence, it gets transcribed, it goes into the cytosol, it gets translated with ORF1 and ORF2, which actually form proteins, that form a ribonuclear protein, which actually translates back or crosses back across the nuclear membrane into the nucleus, and actually can cause a transposition at a different site than where it originated. signs are a little bit interesting as well. Signs actually can be transcribed and translated by ORF1 and ORF2 from line. So it actually requires line expression in order for a sign transposition event to occur. Signs seem to play very important roles in regulating messenger RNA expression. If a line gets inserted into into a sequence that becomes messenger RNA, it can actually bind certain proteins. The ARE-BP protein can actually increase or decrease messenger RNA stability, so it might increase or decrease translation. There are microRNAs that actually bind ALU sequences, so it can actually repress translation that way if it gets inserted. And there are certain proteins called Stauffen proteins. If ALU actually forms a double-stranded RNA, these Stauffen proteins will actually lead to decay. So there's different ways that signs and ALU in particular can actually affect gene transcription and translation. And there's evidence that this has been going on evolutionary for a very long time. There are 48 Mendelian diseases that are clearly related to line 1 transposition. ALU is actually now believed to play a role in both recombination events, but also in genomic rearrangements. When you rearrange the order of the genes on a chromosome, you're actually changing the epigenetic architecture, both with the histone binding proteins and the methylation, so it actually can play a very important role in gene expression. And transposition may even be important evolutionarily. There are 64 proteins that contain translated line 1 sequence. There are 127 proteins that contain translated ALU sequence, and the above doesn't even reflect transpositions that occur in introns or regulatory regions, or de novo transpositions in somatic cells, which are very clearly happening. So, line 1 environmental health. So I'm going to present the small amount of data that Andre and I have actually done on this. The first is going to be on the normative aging study, which is actually a study that we did about 10 years ago, where we just looked at line 1 methylation. That's actually the first study that actually piqued my interest in retrotransposons. And then we did a follow-up study with Heather Burris, in which we actually did line 1 expression in cervical cells in a birth cohort. So we measured line 1 methylation in the normative aging study, which is a cohort of elderly men, and we found that line 1 methylation decreased as patella increased, although it seemed to plateau at a level of about 50 or above. We also found, because we had repeated measures of blood, that line 1 methylation actually increased as you got older. But remember, these guys were 70 to 80. A lot of them who didn't get older died. And so this may actually be a survivor effect, that those who have higher methylation are actually more likely to be the ones to get to 80. So in summary, we found that increased lead exposure in bone was associated with decreased line 1 methylation, and longitudinally, we found that line 1 methylation increased with age, which I think we interpret as a survivor effect, and that higher lead was also associated with earlier mortality. So perhaps lead is actually contributing to some of that reduction of line 1 methylation in those people who die. Obviously, in order to actually tie this all together, we would have to do line 1 expression, which we haven't done yet in the normative aging study, but we have done in the next study I'll talk about. So this study was actually designed by Heather Burris. She wanted to study methylation in cervical cells collected in pregnancy, obviously in pregnant women, and see how that affected their gestational age and other birth outcomes. And so she approached me about doing this study in progress, which is a birth cohort that I lead in Mexico City. And so I thought it was a fantastic idea. So we did it, we collaborated with Andre on this. So we did pap smears on the women in the second trimester. And she actually approached me sort of towards the end of the recruitment phase of progress. So we actually only did the last 80 women that we recruited in the study, and we collected 2 mid-pregnancy cervical swabs, and we got DNA and RNA from those cervical swabs. We did pyrosequencing to determine line 1 methylation, but I was interested in expression, so I talked her into adding it to the microarray that she was doing in nanostring so that we could actually look at line 1 messenger RNA. So these are the results. Heather was very interested in the prostaglandin E2 receptor, which is the top row, but I'm going to ask you to ignore that and just look at the bottom row, which is line 1, and this is line 1 methylation. So we found that line 1 methylation actually predicted fewer days of gestation. So higher methylation was associated with shorter gestational age. But we also looked at expression, and this is where I think it got a little bit interesting. So line 1 methylation actually predicted higher expression, or at least was correlated with higher expression. So remember, these are cross-sectional measurements. So the Y variable in all these analyses is line 1 messenger RNA expression. We also found some evidence that inflammatory enzymes such as TNF, IL-8, and IL-6 were actually also positively associated with line 1 expression. So obviously, the sort of dogma is that higher DNA methylation, lower expression. We found the opposite, actually, that higher DNA methylation, higher expression. There's a couple reasons for that. As I mentioned, when you do DNA methylation in line 1, it's an average of an average of an average. But also, there are a lot of things that regulate protein and gene expression that have nothing to do with DNA methylation. And what we might be seeing is more of a feedback loop where higher expression actually is causing some increased DNA methylation to try to reduce that expression. So as I said, line 1 methylation was positively associated with expression, higher methylation, higher expression. And as I said, suggests methylation may be a reaction to expression and not the primary event. So in conclusion, higher line 1 DNA methylation predicted shorter gestational age. Line 1 DNA methylation was positively associated with line 1 expression. Expression was positively associated with inflammatory cytokine expression. The bottom line is the relationship is very complex. I suspect it has something to do with the feedback loop where methylation is actually reacting to expression. But what I think it also suggests is line 1 may play a role as a mediator or marker of subclinical infection that precedes preterm labor. If you think about it, if line 1 is being expressed in somatic cells and it's inserting itself in its DNA, the most likely thing outside of an inert insertion in a region that is not important is that it's actually affecting gene expression either mildly if it's in a regulatory region or significantly if it gets inserted into an express region, into an exon. So line 1 expression unregulated is almost certainly deleterious to cells on average. So this is our epigenetics team. Roz Wright, Alan Justin, Allison Sanders play a big role in all this. I've never done any epigenetic research without Andrea Baccarelli, so I'm very grateful. And we've had a wonderful relationship over the last 15 years doing a lot of really interesting work. Heather Burris was the brainchild behind the epigenetics work that I presented on cervix. And none of this work could happen without my collaborators in Mexico, Mara Tellez Rojo and Lourdes Schnaz. And that's my last slide. And I apologize if I talked really fast.
Oh, that's perfect, Bob. Thank you so much for such an insightful presentation and reminding us, that some of these repetitive elements are crucial for ring regulation and health as well. So we do have a question from Brenda Kalia. She said, would you expect repetitive element methylation and expression to differ by tissue type? Have you seen this? Both tissue and I guess cells, maybe.
Yeah. I think it will very likely be cell-specific and tissue-specific, just like all epigenetic marks are. And one of the biggest challenges in studying repetitive elements is that they are the same or very, very similar sequence over and over again in the genome, and only a very small number of them are being expressed. Less than 1% are being expressed. So how do you find those ones that are being expressed and try to understand why they're being expressed? That is really a challenge. I think messenger RNA gives us the ability to see that it is expressed, but we're not really seeing where it is in the genome that it's being expressed, because you're going to find this sequence everywhere in the genome. And I think that's one of the biggest challenges.
Right. We have another question from Michelle Hickok from NIHS. She said, have you looked at line 1 methylation and expression in instances of preterm labor? She also asked, how do you think lead is affecting line 1 methylation? So preterm labor, and how is lead affecting line 1 methylation?
So yeah, our study was designed to look at gestational age rather than preterm labor, but I think it would be consistent with the idea that line 1 expression might be a biomarker for preterm labor. I'm not aware that anyone has done that research. I know Heather is interested in doing that going forward. in terms of the question on lead. Lead is clearly associated with lower DNA methylation. I think one of the things that the cervix study illustrates is that it's very hard to make the assumption that higher methylation actually means reduced expression, because what you really have to factor in is the temporal relationship of everything. So if lead directly caused lower methylation, then I would say that it's probably going to cause increased expression. On the other hand, if lead is causing inflammation and has nothing to do with DNA methylation directly, but just causes inflammation, and that causes line 1 expression to increase, you might actually see a change in DNA methylation in response to that. And so it's really hard to know what's happening. You need to know the timing of the exposure, you need to know the DNA methylation, and you need to know the repetitive element expression. And I think when you have all those factors, then you can start to interpret perhaps what's going on. But I think we tend to think two-dimensionally in models that are really three-dimensional. We forget about the temporal relationship. And I don't think our results suggest that, for line 1 at least, that higher methylation means higher expression. I think what it tells us is that the temporality of the DNA methylation change is probably in response to the expression and not driving the expression. That's my interpretation of it, at least. But I think until we get more pieces of the puzzle, it's really hard to interpret these data. And I think we shouldn't just interpret methylation in isolation. I think we have to have expression information and other types of information to really understand what's happening mechanistically.
Great, thank you. Dana Donalino had a comment. She said an exception will be a metastable epiallele like the AVY retrotransposons. These have low inter-tissue variability, but high inner individual variation. Yeah, that's an excellent comment about some of these retrotransposons like the AVY.
Yes.
I did have a question for you, Bob. Has anyone looked at Lyme1 or these alloelements methylation, regulating the epigenome, so methylation of other sites, kind of like doing an EWAS of sign and line to see if there's a relationship there, or looking at the RNA-seq at the same time point to see if there's an interplay between this repetitive element methylation and expression in blood or other tissues?
I've had some discussions with Andrea about how we could try to figure out where in the genome these expressed line 1s are coming from. You're talking about a sequence that's over 6,000 base pairs long, so it's not easy to do sequencing. We never really talked about ALU, though. That's a much shorter sequence. Maybe it might be possible for ALU. I mean, Andrea would know better than me, or Dana, or Carmen, about whether or not the technology allows us to really sequence something that long to try to figure out where it is in the genome.
Great. Well, thank you so much. I think we'll move on. We'll have more time for discussions later. Oh, well, maybe I think someone else submitted a question. What about contribution of DNA hydroxymethylation to repetitive elements? Traditional bisulfite converted DNA methylation. So the question is about DNA hydroxymethylation to the repetitive elements.
That's a great question. I've not seen any literature on hydroxymethylation of repetitive elements. I'm not aware that it's been studied. I could be wrong, but I've not seen anything like that.
Right, yeah, I see Andrea shaking his head. Well, thank you so much, Bob. I really appreciate the presentation, and I hope you're able to stick around for the discussion later on. Next, we have Dr. Lena Colicino. Dr. Lena Colicino is an assistant professor in the Department of Environmental Medicine and Public Health at Mount Sinai School of Medicine. I have had the pleasure of working with Elena, and she's a wonderful biostatistician and environmental biostatistician as well, doing some very innovative work with biomarker development as well. So we're very excited to see some of her latest work, and very grateful that she agreed to present some of this as well. So I see she's still getting set up. So yes, thanks, Elena. I think we can now see your slides. That's great. And if you can just unmute yourself, I think we'll be ready to go.
Thank you so much, Vanessa, for this nice introduction, and also thank you so much for organizing this. I felt a little bit disconnected during this COVID pandemic, and it's very nice being here today. So today I'm going to talk about epigenetic biomarkers of lead exposure, and I've done this analysis under the supervision of Andrea Baccarelli when I was one of his postdocs. So we know lead exposure is a silent disease, has no obvious symptoms, has mostly chronic effects on the cardiorenal system, and leads to adverse reproductive outcomes and cognitive dysfunctions. So lead exposure is a still ongoing issue in the United States, and since passing several industrial regulations and restrictions on lead use, lead exposure has declined in the US, but is still traceable in both adults and children. And we all recall the Flint crisis with contaminated water. It's contaminated water, and the 9/11 tragedy and the contamination of air. But it's mostly an occupational problem. So we have also to say that the adverse health effect of low level of lead can be magnified by other toxicants, and we have recently published on cocaine. So the major sources of lead exposures are paint and its dust, contaminated water, food and soil, but as I said before, is an occupational problem. And we have more than 1 million, mostly men, exposed to a high level of lead. It's worth saying that for every $1 spent on controlling lead hazard, we have at least $17 back in health benefit. Of course, this is not just related to US, it's a more spread problem also to developing countries. So lead enters into the body through ingestion and inhalation, travels in blood to soft tissues, and is partially eliminated by the renal system. But the remaining lead is stored in bones for decades. So the most commonly used biomarker is blood, but blood reflects only 6 months, so a short-term exposure, while cumulative exposure cumulative biomarkers like bones can reflect a longer period. And we have the patella that can reflect up to 20 years, and the tibia can reflect up to 50 years of exposure. So blood lead levels can be tested through blood tests, and we have a threshold that is over which people are constantly monitored, and it's very easy to monitor. But the cumulative biomarkers of lead can be measured only through radiations, and no other threshold is identified, and there's no easy way to monitor, and only few centers have this technology, and it's mostly not used for clinical purposes. So also those type of cumulative exposure can be more appropriate to assess the long-term health effects. So our goal was to develop biomarkers reflecting cumulative exposure of lead that are easy to measure, potentially even safer, so without radiation, that are easy to implement, even retrospectively, and they can contribute to evaluate the long-term health effects. So we are going to use the DNA methylation, which is the most studied epigenetic mechanism, and of course by the addition of a methyl group to a certain base, which is the cytosine followed by the guanine. This is why they are called CPG, and doesn't change the DNA sequence, but has a strong influence on gene activity. We know that it's sensitive to environmental exposure, as we have seen in the previous talk, and the current technology allows us to measure at least methylation levels of at least 500,000 of sites per sample. So we want to use DNA methylation as a surrogate for cumulative lead exposures. So, and we are gonna use the normative aging study, which is a cohort of older men living in the greater Boston area. All of them underwent blood tests, and 350 of them had also X-ray of the patella and tibia. So the statistical approach is in steps. So we have selected the sites that were sensitive to lead exposure through a EWOS analysis. Then we identify lead biomarkers through ElasticNet on a training data set, which consists of 80% of the data. And then we validate those lead biomarkers on the remaining set, which is called testing data, so the remaining 20% of the data. And then we assess the biological pathways of the site mapped to those including in these biomarkers. So people were moderately exposed to lead, and both patella and labia show skewed distribution, so we love to transform both of them. So we selected the sites that were more sensitive to lead, first selecting the sites that were in both Illumina 450K and EPIC platforms, so we can retrospectively measure those biomarkers using 2 different platforms. And we identify about 120 sites that were in patella and 138 in tibia that were nominally associated with lead exposures. And after correcting for multiple comparison, we observed 2 sites that were differentially methylated for their association with patella and tibia lead levels. Okay, so now we want to understand in the training data, so 80% of the data, if those sites that were previously selected jointly contribute to the prediction of cumulative lead levels. And in this case, we identify that 59 sites and 138 sites were jointly contribute to those predictions, respectively in patella and tibia. And we have only 1 site that was in common among those 2 sets. So now we are able to validate using the coefficients that we extract from this elastic net in the validation set. So the remaining 20% of the data in which we have methylation levels. And we are able to estimate lead levels, patella and tibia lead levels, and compare those to actual lead levels. And as you notice, we have a correlation, strong correlation between those 2 measurements. And the accuracy of this biomarker was good for both the models. So we enriched our analysis with the biological pathways, and among the genes mapped to CPG sites that were in the biomarker, we identify an enrichment for Alzheimer's disease and protein digestion and absorptions. And both 2 conditions and diseases were previously identified in relation to lead exposures. So briefly, we have developed and tested 2 lead biomarkers from DNA methylation. And those 2 biomarkers are reflecting 2 temporal cumulative exposure. And those biomarkers are linear combination of sites, and now they can be implemented retrospectively in other cohorts that have the same type of measurements. I want to acknowledge all my collaborators. I have a long list. I couldn't have done this work without them, and also the fundings. And now I'm ready for your questions.
Wonderful. Thank you, Elena. And I don't see any question yet, but I am sure they will come. And I wanted to ask you, meanwhile, about, can you repeat again how you did the validation? Like you took the coefficient from the elastic net, and then you used that same coefficient in the remaining 20%. And from there is where you did to estimate the exposure levels. Can you repeat that part? So that I think it's a complex way of doing. I don't understand it enough myself, so I would love to hear from you again.
So briefly, I have a brief description of this. So basically, at the beginning, we usually do our linear analysis to evaluate the association of few variables with a continuous outcome in this case. But when we have a lot of variables, like we want to select the most important one. Okay, so here are the estimated coefficient with the linear regression. The elastic net that I've used shrink the coefficients towards 0 and set to 0 the coefficients for the variables that are not relevant. So in this case, we have some CPG sites that were selected in the first part, so the EWOS analysis. Then we use this elastic net, and we have all coefficients. Some coefficient will be null, so equal to 0. So they're not relevant for the prediction of this biomarker. Then we use those coefficients, okay? Those coefficients were estimated in 80% of the data set. So the training set. And then we use the 20% that was defined a priori, so we split the data set at the beginning, and this 20% has already methylation levels, right? So in all people, we have methylation levels. So now we can use those coefficients, and we can apply those coefficients to the methylation levels of this new testing set. And now we can reconstruct the exposure levels.
Okay, excellent. Thank you.
Hopefully it's clear.
Yeah, this is very, very useful. And I think it's very important, as we need to do this more, like to move from the elastic net to the...
I had a brief question, Anna, for Elena. Elena, I mean, given your expertise and experience, what do you see as the barrier to come up with these biomarkers, epigenetic biomarkers? You know, we have the aging biomarker, and we have this example of the lead biomarker. Do we need larger sample sizes? Do we need to work, you know, in consortiums to develop these? How do you see us kind of moving forward with this biomarker development?
Thank you so much for this question, Andres. It's a very nice question. And I think the collaborative science can help us to move predictions of exposure predictions, or actually creating novel biomarkers forward. So we can move forward the science with that. I think having multiple cohorts with the same type of epigenetic marks, like in this case, can help us a lot. Because as you notice with the aging biomarker, we have a lot of data. So in this case, I had to split my initial data set in two. So 80% of training and 20% of testing. It would have been nice to have an additional cohort where I could validate these results, or actually doing cross-cohort analysis. I think a cross-cohort analysis can be more helpful in this context. So the major limitation is the sample size, I think.
Great, great. There is one more question, Elena. It's about if there are differences in lead uptake in bone depending on whether the exposure is ingestion versus inhalation. I am happy to help you if you want.
Yes,
that.
Please. Thank you.
So the main difference is that through inhalation, absorption of lead is 100%. We really uptake all the lead that is in the air through the lungs. However, through ingestion, absorption is going to be much more variable, depending on the other metals, on your metals status for calcium, on other microbiome issues, other dietary factors, like if you drink milk or not. So absorption of lead through ingestion is very different. But whatever is ultimately absorbed into the blood, the distribution to the bone will be similar. Good, but I think Elena, it will be very exciting, I think, as we are getting more data, epigenetic data and lead, to start testing those biomarkers. So I think this is incredible work. Thank you. And
Thank
now,
you so much.
Thank you, Elena. And now if you can stop sharing your screen as I introduce our next speaker, who is Maria Argos. And Maria is an associate professor of epidemiology and biostatistics at the School of Public Health at the University of Illinois in Chicago. Maria was also is an alumna of the Mailman School of Public Health at Columbia University, and she was also a trainee of the Columbia Superfund Research Program. She has done a stellar career. She has some of the landmark papers on arsenic and health effects, working with the data from Bangladesh. And it's really exciting that she can share with us some of her work today. So Maria, thank you so much. The screen is yours.
Many thanks, Anna. And I'd like to thank both you and Andres for organizing this mini-symposium and the opportunity to present some of the work that we've done evaluating the association between arsenic exposure and DNA methylation.
And Maria, if you put full screen so that we can see your slides completely, because now we see all the details around. Yeah, great.
Okay, perfect. So to provide some context for this work, I wanted to provide some background for those who may not be familiar with arsenic exposure on arsenic. So arsenic is a naturally occurring metalloid widely distributed in the environment. We know that there's known human exposure to arsenic through various routes, including ingestion of contaminated drinking water, food, soil, dust, and air. However, the consumption of drinking water enriched in inorganic arsenic is really the greatest public health concern globally with respect to arsenic exposure. And the epidemiologic literature to date has widely established that exposure to moderate and high concentrations of arsenic from drinking water is associated with numerous adverse health outcomes, including several cancers. With respect to the global burden of arsenic exposure, the population of Bangladesh has been most severely affected. We know that the groundwater in Bangladesh was discovered to have naturally occurring elevated levels of arsenic, which the population is exposed to through the consumption of well water. Based on data from a well study conducted by the British Geological Survey in the early 2000s, it's estimated that about 50 million individuals in Bangladesh are exposed to well water arsenic at concentrations greater than 10 micrograms per liter, which is the permissible limit suggested by the WHO. And unfortunately, arsenic in groundwater in Bangladesh was only realized after the emergence of an epidemic of skin lesions, at which time the population had already accrued decades of exposure and continues to be exposed today. So I'll be presenting epigenetic data from 2 of our studies in Bangladesh. The first is the Bangladesh vitamin E and selenium trial. This was a randomized controlled trial of 7,000 adults with visible arsenical skin lesions residing in rural regions of Bangladesh. Among those 7,000 participants, we randomly sampled 400 participants for an epigenome-wide association study of arsenic exposure. The data and the samples that we used in the epigenome-wide association study were collected at the time of enrollment. So this was prior to any trial intervention being initiated. Arsenic exposure in the study sample was characterized by measures of blood arsenic and urinary total arsenic concentrations, with the correlation between these two measures being quite high. And we generated epigenome-wide DNA methylation data using the Illumina 450K array. And in addition to having the DNA methylation data available, we had also generated transcriptome-wide gene expression data in the same subsample using an Illumina array. And so we were able to use these data for functional validation in the DNA methylation analyses that we conducted. To give you some sense of the characteristics of the study participants, approximately half of our participants were female. They were all adults. Average age was 43. The average blood arsenic concentration was 9 micrograms per liter, although this varied widely, essentially ranging from 0 to more than 86 micrograms per liter. We also characterized urinary total arsenic concentration, which on average was 300 micrograms per gram creatinine. And again, this ranged quite a bit from 12 to more than 2,000 micrograms per gram creatinine. And as I previously mentioned, all participants in this sample had visible arsenical skin lesions present. We also we evaluated the associations for blood, which are summarized on the left, and urinary arsenic concentrations, which are shown on the right, in relation to whole blood epigenome-wide DNA methylation. In these analyses, we identified 4 differentially methylated loci based on a bond-for-only significance threshold. And the associations that we observed were consistent between the 2 exposure metrics. With respect to the biological functions of these identified methylation sites, these genes have been implicated in skin carcinogenesis as well as hypertension. Both these endpoints are relevant for chronic arsenic exposure, and the associations with skin carcinogenesis are particularly interesting since the analyses were conducted in a sample which is at elevated increased risk for eventually developing skin cancer, since they have the presence of skin lesions already. We used the existing gene expression data on the same study participants to conduct functional validation of the identified CPG sites. And so we looked at transcript levels of the corresponding genes from the CPG sites identified. In those analyses, we observed a positive association between the CPG site in PLA2G2C with mRNA transcript levels of the gene, as well as a positive association of the CPG site in SLC4A4 with its corresponding mRNA transcript levels. And unfortunately, for the remaining 2 CPG sites, we did not have expression probes available for those corresponding genes and be able to look at the functional effects of those methylation sites. We also recently generated DNA methylation data on participants from the health effects of arsenic longitudinal study. This is a long-standing population-based cohort, which has been funded through the Columbia Superfund Research Program to evaluate the health effects of chronic arsenic exposure in relation to a wide array of health outcomes. Among the more than 12,000 adult participants that were originally enrolled in that cohort, we randomly sampled 396 participants for an epigenome-wide association of arsenic exposure. And in this population, arsenic exposure was characterized by urinary total arsenic concentration, as well as well water arsenic concentration of the primary drinking water source. The correlation between these 2 exposure measures is a bit more moderate. And within this sample, we used the newer Illumina EPIC array to generate epigenome-wide DNA methylation data. So this provides data on nearly 900,000 CPG sites. With respect to the sample characteristics in this population, a bit more than half were female. The average age was 36 years. The average urinary arsenic concentration was just a bit lower at 200 micrograms per gram creatinine, with very wide variability among the sample. The average water arsenic concentration was 50 micrograms per liter, so notably higher than the level recommended by the World Health Organization, but there was also wide variability with respect to the water arsenic exposure. And the major contrast between the participants in the HEAL sample versus the study that I just described with participants from the BEST sample was that the participants in HEAL had not developed arsenical skin lesions. So that's the major distinguishing characteristic between these two studies. So in the discovery analyses using the 396 HEAL participants, 7 CPG sites were identified to be differentially methylated with respect to urinary total arsenic concentration. So this was based on a Bonferroni significance threshold as indicated by the solid gray line in the figure. And then when we considered an FDR threshold of 0.05, as shown by the dashed gray line in the figure, there were 34 CPG sites that were differentially methylated. A large proportion of the identified CPG sites that were available on the 450K array replicated based on a nominal significance threshold, and they were directionally consistent when we looked back at the original BEST sample. We also had the opportunity to conduct a meta-analysis combining data from the 396 participants from HEAL and the 400 participants from BEST, using approximately 390,000 common CPG sites between the two Illumina platforms. So this doubled the sample size of our original analyses for urinary total arsenic concentration. And in this combined analysis, we observed 41 associated CPG sites based on the Bonferroni significance threshold and 221 CPG sites based on the FDR threshold. When we looked at enrichment for these genes, there was significant enrichment of genes annotating to reactive oxygen species pathways, inflammatory response, and tumor necrosis factor alpha and nuclear factor kappa B signaling. So with respect to the extensive literature that's been published to date, looking at the association between arsenic and exposure and DNA methylation, it's actually been very challenging to synthesize the literature. So these are some considerations looking at what's been published to date. There's considerable variability when we look at how arsenic exposure was assessed in these various studies. So with respect to timing of exposure, whether it was prenatal, concurrent to when DNA methylation was measured, or if it was assessed longitudinally with respect to DNA methylation at some future point. How it was assessed also varies widely. So as observed even in our studies, we've used various exposure metrics for arsenic, which makes it difficult to synthesize results across the literature. And then the levels of exposure also dramatically vary across study samples. So with respect to the populations that I presented, these populations have moderate to high levels of exposure, where there are a number of studies that have also published in very low exposure populations. There's also many differences with respect to the genetic ancestry of the various study populations that have been examined, making integration of these observations difficult due to potential gene-environment interactions. And as seen with our studies and other published studies, the disease status of the population varies, which may impact the observed DNA methylation patterns. There's notable differences with respect to statistical approaches in processing and analyzing the data. Few of these studies have examined sex-specific differences, nor have they really been sufficiently powered to look at associations disaggregated by sex, so this should be a consideration moving forward. And finally, many studies have looked at either DNA methylation in venous blood in adults or within the context of prenatal exposure that we'll be hearing about umbilical cord blood methylation. But few studies, I think actually just one to date, has actually evaluated methylation in other target tissues, which I think will be an important consideration for characterizing the epigenetic effects of arsenic. So I'll conclude by acknowledging the large study team that's contributed to this work to date. And if there's time, I'm happy to take any questions.
Thank you, Maria. That was great. So feel free to submit your questions on the chat box. I did have kind of like a bigger picture question for you in terms of thinking on the health effects of arsenic and thinking about the lacks for some of the cancers that people have documented. Do you think, I mean, in terms of collecting biomarkers, and a lot of us are doing urine and other things. You mentioned the correlation between urine and blood. Do you think urine might be the best biomarkers in terms of epigenetic effects, or should we be thinking about things like hair or nails or something that reflects a longer term exposure?
Yeah, that's a great question. So I think in the context of our studies where we know that there's daily chronic exposure to arsenic through drinking water, urinary arsenic probably characterizes this exposure very well. In other populations where exposure may not be daily, so perhaps from food sources or other sources, we might want to consider looking at toenail arsenic, which is a more integrated, longer term exposure marker for those populations. So I think to some extent that may be a bit population specific depending on what the source of exposure would be. In terms of what exposure construct to look at, I think, one interesting question would be to potentially look at associations with urinary metabolites. We know that not all individuals metabolize arsenic with the same efficiency. And so those who are less efficient metabolizers of arsenic may be at increased risk for arsenic toxicity. And to date, there have been very few studies that have actually looked at arsenic metabolites with respect to DNA methylation. So I think, that potentially may reveal some interesting effects of arsenic's effects on the epigenome with respect to individuals who may be the most vulnerable to arsenic exposure.
Thank you. We have a really interesting question here for Corin Mann. They mentioned, is there a possibility to assess the immune microenvironment of the skin lesion? So actually thinking about the target tissue to see if it correlates with methylation, gene expression, or inflammatory markers observed, i.e. is there lesion tissue collected? I don't know if you're aware of anyone collecting skin lesion tissue. I think this is a great idea, but maybe you can speak about this, whether skin lesion tissue we might be able to collect, or if anyone else has looked at it.
Yeah, that's a great comment. So actually within the context of the Bangladesh vitamin E and selenium trial, for individuals who over the course of the trial developed what was expected to be a non-melanoma skin cancer, those lesions were biopsied. And so within a subset of the trial participants, we do have skin tissue available. We haven't yet analyzed those tissues within the context of the methylation data, but that is a very interesting consideration. I'm not sure if you know of other studies that have skin tissue. I think maybe not many others.
Yeah, I'm not sure either, but that's a really great suggestion. Thank you. We have another question about which areas in Bangladesh do you study? So I guess if you can be specific about the two areas in the two studies, and what are the inclusion criteria for the subjects? If you can expand on that a little bit.
Sure. So for the HEAL study, that is in a rural area known as Arihazar. It's located directly west of the capital city, Dhaka, about 2 hours west of the capital city. For the Bangladesh vitamin E and selenium trial, that was also conducted in Arihazar, but also in collaboration with ICDDRB. We expanded to another rural area in the southwest of the country known as Matlab. So that's the main area that ICDDRB has been very active in with respect to looking at arsenic exposure in the population. So those were our two study areas. With respect to inclusion criteria of the HEAL cohort, participants were required to be adults, 18 or older, and reporting consumption of drinking water from a tube well within our defined geographic area. Those were essentially the main criteria, and had resided in the area for, I think, at least 5 or more years. Within the Bangladesh Vitamin and Selenium trial, again, these were adult participants, but the age criteria was a bit more narrow. So these were individuals between the ages of 25 and 65 years old. And the other criteria was that they, upon visual examination, had presence of arsenical skin lesions.
Thank you, Maria. There's one more question about what is known about the methylation status of arsenic in the urine, and I'm not sure if Kiran is referring to the different species. So maybe you can talk a little bit about the different, methylated arsenic species in the urine, perhaps. Okay, yeah, she said yes. So maybe you can refer that maybe specifically to your study. And also, if you could get closer to the mic, it was a little bit lower in the last response. So there we go. I think that's.
Sure. So actually, there's been quite a bit of work characterizing individual variability with respect to arsenic metabolism. Several groups have identified genetic variants in the arsenic methyltransferase gene. So this is AS3MT, where genetic variants in that gene may affect how individuals metabolize arsenic once it's been ingested. And so with respect to exposure biomarkers, this variability can be characterized by measuring specific arsenic species in the urine. So what's typically measured is inorganic arsenic, dimethylated arsenic, DMA, and monomethylated arsenic, MMA. And what's been typically observed is that individuals who have a larger proportion of monomethylated arsenic in their urine may be at enhanced risk of developing essentially a whole host of arsenic-related outcomes. So development of skin lesions, cardiovascular, cardiometabolic diseases, and cancers.
Great. Thank you so much, Maria. I think we don't have any more questions. And in the interest of time, I will move on. If you don't mind, stop sharing your screen, and then maybe Anne can start sharing her screen. It is my pleasure to introduce Dr. Anne Bosak. She's a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai. She was a trainee at the Columbia Superfund Research Program, and I had the pleasure of working with her as well as she came to the Oregon State Superfund Research Program on an externship. And she's done work really throughout the life course. So the title of her presentation is Arsenic and DNA Methylation Across the Lifespan. So thank you, Anne, for presenting, and please take it away. Sorry, I think you might be muted. So yeah, there we go. Not yet.
Okay, I think that worked, yeah. And you can see my screen? Okay, perfect. So as Andre said, most of this work was done while I was at Columbia, but I'm currently a postdoc at Mount Sinai. That's why there's the 2 logos there on my presentation. So my presentation will actually focus on 2 studies. The first one looking at in utero arsenic exposure and birth outcomes and mediation by DNA methylation. And the second one is an epigenome-wide association study conducted in adults. So probably most people on this call are aware that arsenic exposure is associated with a broad range of health effects, including adverse birth outcomes and health effects in adults affecting most organ systems. And the health effects associated with arsenic exposure persist decades after exposure has ended or been reduced. So this suggests that epigenetic dysregulation may be a mechanistic link between arsenic exposure and health outcomes. During the time of embryogenesis, extensive epigenetic reprogramming occurs. So environmental exposures during this time may be particularly dysregulating to the epigenome and results in health effects. So the first study is focusing on in utero exposure to arsenic and epigenetic dysregulation as a mediator between arsenic exposure and adverse birth outcomes. This study was previously published in Epigenetics. So here's a screenshot of the article if anyone wants to go back and reference it. This was conducted with Dr. Molly Kyle at Oregon State University as part of a Superfund externship. So the overall objectives of this study were to investigate associations between in utero arsenic exposure and birth outcomes and mediation by DNA methylation. And we use both a discovery and validation approach, which I'll talk more about in a couple of slides. This study was conducted in a prospective birth cohort in Bangladesh. And arsenic was measured in drinking water and maternal toenail samples, and DNA methylation was measured in cord blood in 44 participants using the 450K microarray and using pyrosequencing in a larger sample of almost 600 participants. So as I mentioned, we used both a discovery and validation approach to study mediation. Using the discovery approach, we conducted an EWAS to identify potential sites that mediated the association between in utero arsenic exposure and birth outcomes, and identified 3 targets located in MIR124-3, GNAL, and MCC. DNA methylation in these genes were further measured using pyrosequencing in a larger sample. So I'll focus on those analyses in the pyrosequencing data. Our statistical approach was used to use structural equation models, or SEMs. This is a statistical approach that uses factors of factor analysis, regression, and path analysis. This is useful for this type of data because it allows you to use multiple indicators of the same construct and indicators that are highly collinear as well. So when you measure DNA methylation using pyrosequencing, you obtain data not just of the target site, but also of neighboring CPG sites and possibly SNPs if there are SNPs in the area. So that's potentially very helpful when conducting this type of analysis. And in the case of our data, DNA methylation of each of the pyrosequenced genes was highly correlated, which really lent to the structural equation modeling approach. So in this approach, we created a latent variable using DNA methylation measured at all of the CPG sites in that region. And then this latent variable was used in a structural equation model to assess mediation of the association between arsenic exposure and gestational age by this latent variable. So we created structural equation models for each of the 3 target genes and observed a positive association between arsenic exposure and DNA methylation of the latent variable. We observed significant mediation for MIR124-3 and MCC between arsenic exposure and gestational age by DNA methylation. However, we did not observe significant mediation with GNAL. So we took these 2 genes, MIR124-3 and MCC, and created a single structural equation model to assess mediation. And you can see this is kind of a complicated diagram when you start to put in all the CPG sites as well as potential covariates. So I'm just going to zoom in on the most important part. So here we observed mediation of DNA methylation by DNA methylation of MIR124-3 of the association between arsenic exposure prenatally and gestational age. This mediation pathway was achieved a p-value of 0.06, so marginally significant, but in sensitivity analyses in which we use maternal toenail arsenic instead of water arsenic, this achieves significance at 0.05. So to sum up this study, epigenetic dysregulation may mediate the association between in utero arsenic exposure and birth outcomes. So using a discovery and validation approach, we observed that DNA methylation of miR-124 mediated this association. miR-124-3 is potentially a biologically relevant gene. This is a microRNA gene, and it has been associated with arsenic exposure in mice, and has also been associated with birth weight in mice. So next I'm going to move on to the epigenome-wide association study conducted in adults. And Maria Argos already gave a pretty good overview of previous EWAS conducted with arsenic exposure, but I just want to summarize them again in the context of this study. So this table just shows the previous EWAS of arsenic exposure. And as you can see here in the summary of findings in the last column, there's a broad range of outcomes. In some cases, no significant CPG sites were identified, in some cases in the hundreds, and in one study, almost 5,000 significant hits were found. So it's really hard to compare outcomes of cross studies, and in part because of the differences in methods that Maria mentioned. I also want to point out that a lot of these studies were conducted in birth cohorts, and they also had relatively small sample sizes, so that definitely impacts power when you're working with these large data sets. So in this study, and I just want to briefly mention that this study is currently in press, so I should have a screenshot of the PDF hopefully very soon. So this study was conducted in adults with low to moderate levels of arsenic exposure. And we identified both differentially methylated positions and differentially methylated regions associated with total urinary arsenic levels. The study was conducted in the Strong Heart study, which is a population-based prospective cohort in 13 US American Indian communities. We used data from the phase 1 baseline exam, which was actually conducted in 1989 to 1991. So about 30 years ago, these blood samples were collected. DNA methylation was measured using the Illumina methylation epic array, which measures DNA methylation at over 850,000 CPGs. This slide gives an overview of the sample and data processing. I won't get into the details of this, but I'm happy to talk about it in the questions or offline. But we had a final sample size of 2,325 participants, and over 780,000 probes were analyzed. This is a volcano plot of results from our analysis of differentially methylated positions. The dotted line shows the FDR level of significance, and the solid line shows the Bonferroni level. So in summary, we found 20 sites that were significant at the FDR level and 5 significant at the Bonferroni level. This table shows the 20 sites that were significant at FDR. I can't go through all of them right now, but I do want to point out a couple of things in this list of sites. Overall, we saw a positive association between DNA methylation and arsenic exposure, with exception of 2 of the top 20 hits. We also saw this positive association in the volcano plot, which shows all of the associations tested. So as you can see, the points to the right of 0 represent a positive association between arsenic exposure and DNA methylation. So there's this trend towards increased DNA methylation with arsenic exposure overall. Among these top hits, I want to point out a few that potentially have biological relevance. Our top hit was located in SLC7A11, and this gene is involved in the cysteine glutamate antiporter system, which is involved in GSH biosynthesis. GSH is an antioxidant, which is potentially protective against arsenic-induced oxidative stress. We also identified 2 significant sites in LINGO3, which has previously been identified as a gene hub in melanoma tumors. And 2 sites located in CSNK1D. And this gene has been associated with a broad range of cellular processes, including DNA replication and apoptosis. And prenatal exposure to arsenic has previously been associated with DNA methylation and gene expression. We also identified one differentially methylated region located in chromosome 11. This region included both an open chromosome reading frame and the gene G-span 32. So this plot just shows the location of the gene of the region on chromosome 11, as well as the differentially methylated region shaded in gray. So I'm just going to zoom in on that region. Consistent with our overall findings, there was a positive association between DNA methylation at this region and arsenic exposure. There's not much research regarding this area of the genome. However, genetic variation has been associated with Beckman-Weinman syndrome, as well as leukemia. So just to summarize the findings of this EWAS, this is the largest EWAS of arsenic exposure to date, and it's the second to use the A50K microarray, which is advantageous because it has increased coverage of regulatory elements, as well as increased coverage of the genome overall. And we found that total urinary arsenic levels were associated with both differentially methylated positions and a differentially methylated region. Oh, sorry about this typo on the slide. But just to summarize, overall, we found that the association between in utero arsenic exposure and birth outcomes is possibly mediated by changes in DNA methylation. And arsenic exposure in adults was associated with differential DNA methylation. In both of these cases, more research is needed to fully understand how arsenic exposure affects specific genes and CPG sites, and how that's translated into changes in gene expression, and in turn translated into health outcomes. So just to conclude, I want to thank, of course, all of the study participants, both in Bangladesh and in the US. Both of these studies combined include about 3,000 participants. So that's definitely a lot of people to get involved in the research, and couldn't be done without the study participants. I want to thank Anna Navasasian's lab, who I worked with in the Strong Heart study, Molly Kyle's lab at OSU, who I worked with for the Bangladesh study, and Mary Gamble's lab, who is was my mentor at Columbia when I was doing my PhD, and of course the Columbia SRP, and Andres and Anna for organizing this meeting.
Wonderful. Thank you, Anne. And let's see if we have questions coming. I am sure there will be some questions. I have a couple of questions to you. The first one is about the microRNA gene that you found signal potentially in your first study. And I was wondering if that could put some of this connection with the work that Carmen Marcet was mentioned at the beginning. Do you think this could be related, some of these DNA methylation signals related to some microRNA genes?
Yeah, I think that's a really interesting connection, because DNA methylation and microRNAs are both kind of classified as epigenetics. I mean, some people might disagree with microRNAs, but as my work actually is shifting more to microRNAs in my postdoc, I'm starting to think about how in turn epigenetics may regulate microRNA expression. And so, yeah, definitely it would be really great to have a cohort in which we could look at both DNA methylation and microRNA expression and see how those are associated.
Very good. My second question is related to old methods and new methods, statistical methods. So the statistical method seems to be continuously changing. And I wonder if you, and I know you've done some more multidimensional methods to look at the EWAS analysis in the Strong Heart, even if you didn't mention it here. But do you think it's going to be important for us to revisit like old studies or old analyses using new statistical methods? Is that worth the effort? Since we have a number of studies that have data, should we reanalyze?
Yeah, no, I think that would be great. I mean, anyone who works with the 450K and 850K microarray data knows it's expensive to run, and it's hard to recruit these really large cohorts. So there's a lot of data out there, and as the statistical techniques progress, I think it would be great to be able to go back and reanalyze data in different ways and be able to pull more information from it.
Very good. And then my last question, and then I'll see if somebody else has more questions, is can you share a little bit your experience of translating this complex data with participants and communities?
Yeah, this was actually like a really nice opportunity to focus more on the science or the analysis and outcomes, because in a lot of my presentations, I spend a good amount of the presentation trying to provide background information and explain what DNA methylation is and epigenetics is. So I've thought a lot about how to simplify that information. And in the Strong Heart study, what Anna's referring to is there's a newsletter that goes to participants and summarizes the findings, and just kind of increases buy-in from participants if they know where the data is going and why it's important. So we put together a summary of DNA methylation and epigenetics and why we're collecting these samples and doing these analyses to explain that to participants. And I think that's really crucial to buy in, especially in these large cohorts with a really long follow-up period. So participants are able to understand what's going on with their data and just kind of give them some background to the science rather than just going and donating blood and urine every so often and answering a lot of questions.
Very good, thank you. So I don't see any other questions. So please, if you have questions, feel free to email or contact Anne. And so now it's my pleasure to introduce the next speaker. So Anne, if you can stop sharing so that Arthe can start sharing her slides. So our next speaker is Arthe Domingo, and she's currently a PhD student at the National Institute Center for Epidemiology in Spain. And she, until very recently, she was a data analyst working with us here at Columbia University. She's a mathematician and a biostatistician, and she worked with us for the last couple of years, and she's continuing to work with us as she's moving into her PhD, mentored by Maria Telles Plaza at the National Center for Epidemiology in Spain. And I wanted to say that Arthe has been a tremendous contribution to the epigenetic work in the Strong Heart study. None of the work that we are now doing would be possible without her. She has really brought tremendous expertise, knowledge, dedication, tremendous hard work. And I was very worried because we were transitioning from Hopkins to Columbia, lots of changes in the team, and suddenly Arthe came, and I am so thankful, Arthe, to your very hard work, and thank you for coming today and sharing some of the things you've done.
Thank you, Anna, for this very kind introduction. And thank you all for being here today. I wasn't expecting so many people. So I will be talking about two projects today. One was recently published, and the other one is still ongoing at a very preliminary stage, but I just wanted to share with you some interesting results that I think are interesting to share. So smoking is the leading cause of preventable death. Declines in the number of smokers have been reported by the World Health Organization in the last years. However, 1.3 billion of people will still be smokers by 2025, according to predictions. Smoking is also the second risk factor, only after high blood pressure, responsible for the highest number of deaths in the world. So smoking has been consistently associated with epigenetic dysregulations in concrete genes across multiple populations. One of the main toxicants in tobacco is cadmium, It's associated with smoking-related health outcomes such as cardiovascular disease and cancer. And it has also been associated with changes in global DNA methylation. However, the epidemiological studies in blood DNA methylation and cadmium are scarce. So we hypothesized that cadmium might be partially mediating the association between smoking and DNA methylation. And this leads to the first project that I'll be talking about, which is the association of both smoking and cadmium with DNA methylation, as well as the potential mediating role of cadmium on the association between smoking and DNA methylation. Now, as we all know, one of the main health effects of smoking is cancer. Smoking is associated to 14 different types of cancer, and 36% of smoking-related deaths are attributable to cancer. The main type is lung cancer, as smoking is responsible for 80 to 90% of lung cancer deaths. One of the biological mechanisms by which smoking could contribute to the development of cancer might be DNA methylation, as we know that it changes healthy regulation of gene expression to a disease pattern, and also DNA methylation changes occur in early stages of tumorigenesis. So this leads to the second project that I'll be talking about, which is the potential mediating role of DNA methylation on the association between smoking and cancer. We are looking at all smoking-related cancers, but I'll be focusing in lung cancer due to the lack of time. And as I said before, this is still a very preliminary work. So to develop these projects, we use data from the Strong Heart study. This is the same study that Anne was talking about in the previous talk. It's a population-based prospective cohort supported by the National Heart, Lung and Blood Institute and the National Institute of Environmental Health Sciences. It includes 12 American Indian tribes, as Anne said, and more than 4,500 participants. And it has cancer incidence follow-up for almost 30 years. So in this chart, we can see an overview of the different visits of the study. And DNA methylation was measured in 2,351 participants at the baseline visit. So Anne already showed the exclusions that applied to this data, but I'm just going to summarize the pre-processing a little bit. DNA methylation was measured using Illumina's methylation EPIC bed chip, or 850K. Samples with low intensities or non-bimodal distributions and CPGs that were technical failures were excluded. We applied both single sample NUC normalization for background correction and regression in correlated probes normalization to account for probe type bias. Sex chromosomes, cross-hybridizing probes, and SNPs with minor allele frequency greater than 0.05 were excluded. And we also applied batch effect correction. So after all these pre-processing and exclusions, we had 2,325 participants, as well as almost 800,000 CPGs for the analysis. So I'm now going to talk about the first project, smoking, cadmium, and DNA methylation. This work was published some weeks ago in Environmental Health Perspectives. We did epigenome-wide association studies using the R package Lima, which conducts regressions in each of the CPG sites. We did this for cadmium. This is urinary cadmium, by the way, in both overall and never smokers, and also for smoking, current versus never smokers, former versus never smokers, and cigarette pack years or cumulative smoking dose. For the mediation analysis, I will focus in current smoking. We used beta regression additive models, as well as the product of coefficients method to obtain total effects, indirect effects, and direct effects. So here we have the number of differentially methylated positions for all exposures. We can see that current smoking has many more. And also 4 of the 6 differentially methylated positions for cadmium are still significant when restricting the analysis to never smokers. So these are the top genes in the cadmium EWOS. And now I'm bolding the ones that are still significant when restricting the analysis to never smokers. So we can see that the two only ones that are not are in this region of chromosome 2 that has been reported many times as associated to smoking, but the biological function is not known. These are the top genes for the current smoking. All these genes are very well known in the smoking and DNA methylation literature. And we can see that these FDR-corrected p-values are much more extreme than the ones for cadmium. Regarding the mediation analysis, here I'm reporting the total effect of smoking in DNA methylation, the mediated effect through cadmium, and the relative mediated effect, which is the percentage of the total effect of smoking in DNA methylation that is attributable to cadmium. So as we can see for the first CpG, for example, the relative mediated effect is about 13%. So as conclusions, we replicated the association of smoking-related genes and also found new ones, as this is the first study using the EPIC array to assess the relationship between smoking and DNA methylation. Cadmium was associated to changes in DNA methylation even in never smokers. The effect of smoking in DNA methylation might be partially mediated by cadmium. However, most of the effect is still unexplained, so further research regarding other toxicants of tobacco is needed. And this is the screenshot of the paper in case someone is interested in reading it.
Thank you. Oh, sorry. Go ahead.
So now I'm going to talk a little bit about the second project, smoking, DNA methylation, and lung cancer. In the previous work, we considered the CpG sites one by one. However, when we kept working with this data, we realized that it would be more appropriate to model them all together, as they are highly spatially correlated and there is a huge confounding between CpG sites. So it would be more appropriate to consider the effect of the CpG sites in presence of the others rather than modeling them individually, as Elena was talking about before. So we did an epigenome-wide screening to select the CpG sites that were related to lung cancer. We used adaptive elastic net, which is an adaptive version of the tool that Elena explained before, and it allows to introduce CpG sites all together in the same model. For the mediation analysis, we used Allen additive hazard models, which are the standard tool for mediation analysis in a survival setting. So we found 35 differentially methylated positions associated with lung cancer, and we did the mediation analysis individually in each of those CpGs. Here I'm showing the top ones. Again, I'm showing the total effect of smoking in lung cancer, the mediated effect through DNA methylation, and the relative mediated effect, which will be the percentage of the total effect of smoking in lung cancer attributable to DNA methylation. So as we can see, the top CpG here has about 81% of relative mediated effect, which will mean most of the effect of smoking in lung cancer mediated by these by methylation changes in this CpG. However, I have to say that these mediation models are not independent, of course, because the CpG sites are correlated and also the biological pathways are intertwined. So multimediator model in this setting would be challenging. So again, this is very preliminary, but a large extent of the effect of smoking in lung cancer might be mediated by DNA methylation changes in well-known smoking-related genes. However, these results need to be interpreted with caution, as in the cancer setting, oncogenic transformation might be ongoing for many years before the cancer is actually diagnosed. So the biological interpretation is complicated because we don't know if at the time of the measurement of DNA methylation, these oncogenic transformation were already ongoing. So to finish, this is a summary of all the ongoing projects with the DNA methylation data of the Strong Heart study. These data have a huge potential as we have a big sample size in a big microarray, long follow-up for several disease outcomes, as well as very rich information in lifestyle and socio-demographic variables. So this is a great opportunity to learn from these data and from these communities. And I just wanted to thank all the collaborators of the studies, and especially Drs. Navas Acien and Tellez Plaza, who have been my mentors, as well as all our funding sources that are listed below. And I'll be happy to take any questions.
Thank you, Arce. That was really nice. I apologize for interrupting a little bit earlier today. I was fascinated by your results in mediation and cancer, so we're still waiting for some questions to come in. But I wanted to ask you about, sometimes when we submit papers and grants, and I think these are our colleagues that are more in the, basic bench science, thinking about mechanisms versus the causal mediation analysis that you have been doing. And as you know, given that methylation is influenced throughout time and environment as well, do you see this as competing interests? You perform a causal mediation analysis and you have some really interesting results for cancer, but as you also mentioned, you have the potential for maybe reverse causality. So how is there a way that we can kind of resolve this type of two competing interests of doing, explaining mechanisms, but also finding biomarkers, and especially for the AHRR signal that you found to be so consistent that with smoking, and now you have shown that this is due to cadmium, potentially some of it as well.
Mm-hmm. Yeah, so to avoid this reverse causation issue, a Mendelian randomization study, for example, would be appropriate. There has been actually a Mendelian randomization study focused on the gene AHRR with lung cancer, and they did not find, in fact, any causal role. However, that sample size was small, and, I think more studies are needed in these lines. There's definitely something going on, with AHRR, because it has been, there are other studies that have actually done, formal mediation analysis, and they have found these associations. But still, as long as you don't do, longitudinal analysis or Mendelian randomization, you always will have this limitation of the reverse causation issue. So yeah, I think this is challenging. Repeated measures of DNA methylation potentially, but for cancer in concrete is more challenging because, we don't know when this actually started, so we don't know if it predates or not. So.
Perfect. We have another question by Corinne Mann. Does your data suggest that the AHRR might play a role in cadmium-mediated carcinogenic mechanisms beyond the role that is predicted in mediating the effects of polycyclic aromatic hydrocarbons?
If my data suggests that?
Yeah, if it's potentially, beyond what is predicted by the PAHs, I guess. Yeah. In terms of maybe the smoking.
Mm-hmm. So I haven't done any research on that, and unfortunately in the Strong Heart study we don't have exposure data other than metals. But I think more studies are needed to investigate the other toxicants, because cadmium was shown to play some role, maybe to mediate some of the effect, but the effect was not really huge. So a lot of the effect is unexplained. So it would be nice to do an analysis of, well, not all the toxicants, because I think there are 7,000 or so, but the main toxicants of tobacco, and to see the different mediating roles of them and compare.
Great. I had another question about if you expect the cadmium to be mostly from smoking in this population, or are there any other sources that you might suspect might be contributing to the biomarkers as well?
So in this population, we expect it mostly to be from smoking, but there is another source in the Strong Heart study, although it's much less than smoking, which is processed meat. In fact, there is a paper from one of our collaborators which states that in fact, people have cadmium in their bodies coming from processed meat, but again, it's less than from smoking. And also cadmium sometimes might come from occupational exposure, But this is typically, for example, minery, which in the strong heart study is not the case. And also it could be jewelry, for example. There might be some people in the strong heart study are working with this manual, jewelry and getting some cadmium from there. But I think the main source is definitely smoking, as this is a population that there are a lot of smokers and also, secondhand smoke and everything. So yeah, it's definitely the main source.
Is that cat on the bed for his morning nap?
Excuse me?
Oh, sorry, I think someone was unmuted and they were talking.
Okay. I wanted to briefly ask you, because with these causality issues and all of that, and I know you are very interested in prediction beyond causality. So maybe, do you think this signal, this very strong signal of 80% of the smoking lung cancer predicted by this CPG, could that be useful for prediction purposes beyond what is the causal role of this HAR in lung cancer development?
Yes, definitely. Actually, I didn't talk about this, but I ran an this adaptive elastic net model for lung cancer. It wasn't only in the AHRR gene, but in the whole array. And I got a C index of like 90%. However, I mean, this needs to be replicated, of course, in external populations, because this wasn't even, it was like one population and no training or test sets or anything. But there have been other studies reporting that AHRR might be very useful as a biomarker for early detection of lung cancer. So I think, yeah, this has a lot of potential for prediction, maybe even more than for causality at this moment, because we don't have, longitudinal data and so on. But for prediction, definitely, yeah.
Well, wonderful. Thank you so much, Arce. I don't see any other questions for now, but please stay online. Maybe people will have other questions. Sure. Next, it is my pleasure to introduce Dr. Kelly Bakolski. She's an assistant professor at the University of Michigan School of Public Health. And anyone that has worked in birth cohorts and as well as doing these EWASs is very appreciative of Kelly's major contributions to develop a cord blood reference panel. I'm sure I am, and many of our collaborators in PACE are as well. So Kelly has really revolutionized the field, but really contributing to these cord blood reference panels as well as other methodological work to help us better interpret these EWASs. So we're looking forward for her presentation titled Pregnancy and Perinatal Metals Exposures on the Epigenome. So Kelly, thanks so much for your participation, and please take it away. Sorry, I think you're muted. There we go. Yeah.
Thank you. As Andres said, my name is Kelly Bakolski, and I'm a junior faculty member at Michigan. And I want to thank Anna and Andres for your leadership and vision in organizing the symposium. I think you can tell from the attendance and the excellent audience engagement that you're filling a need for scientific discussion and connection during this period that can be isolating. So I'm thrilled to participate. Today I'm going to share with you two stories of investigation of metals exposure in epigenetics. The first focuses on pregnant mothers with an epidemiology design, and the second looks at the effects in offspring from perinatal exposures with a toxicologic experimental design. So this first project I'm going to share with you was led by Dr. Max Ong, who's a postdoctoral fellow and a really excellent quantitative scientist and creative scientist. It was recently pushed to medRxiv, so you can look to this for more investigation, and Max is on the faculty job market. He is an outstanding candidate and collaborator. So as we know, the uniting principle that's bringing all of us together today is this issue that trace metals exposure is widespread in our environments, in our homes, in our lives. And appropriately so, we often think about the effects of these exposures on vulnerable developing fetuses, but I want to acknowledge today that this also is an important time period in the health of the mother, and pregnancy may be a vulnerable period for women's health. So to that end, we looked at maternal exposure to 5 metals, 3 toxic, what we would consider toxic metals in all circumstances, cadmium, lead, and mercury, and 2 essential metals, manganese and selenium, measured in pregnant mothers during early pregnancy in their blood. And we tested for association with DNA methylation concurrently, so also during early pregnancy in the blood of the mothers. This project was done in the context of the Early Autism Risk Longitudinal Investigation. It's a prospective pregnancy cohort design. The mothers were tended to be in their 30s. They tended to be non-Hispanic white. They were highly educated. They had higher incomes, and their offspring were equally likely to be of either sex. When we think about the exposure levels in this population, they were detectable in this population. And when we compare the exposures to, in this population to those observed in NHANES among women of reproductive ages, we see that our population's in navy, NHANES is in orange, and we see that they're fairly similar. So at least in this respect, the findings may be generalizable to a US population. And next we looked at each metal individually with methylation across all of the sites individually on the Illumina 450K methylation array, which has been discussed at length today as a useful tool. So just by way of visualizing and understanding what are the general associations observed across each of these metals, we can count how many sites reach different p-value or significance thresholds by each metal. And what we observed is that lead and manganese in our population had the highest proportion of sites associated with DNA methylation. So I'll focus on those two here. So first, looking at DNA methylation sites associated with manganese, on the x-axis I have the difference in percent methylation per interquartile range increase in manganese observed. We see that there are several sites associated with increased methylation in this population. And when we try to look for patterns across those sites associated with manganese, so in the form of biologic pathways enriched, we observe that there's enrichment in kind of fundamental biology and cell cycle pathways. So cell cycle processing, nucleic acid metabolism, cellular nitrogen processing, which is related. So there's some foundational biology in terms of the gene targets of manganese. Similarly, we highlight the sites associated with lead exposure in our population. These, again, we saw all lead exposure here is a percent methylation per natural log increase in lead was associated with hypermethylation at all of these sites. And some of the sites of interest, we noticed there's cytochrome P450 enzymes, so important biotransformation enzymes. There's important developmental enzymes, including BMP and Hox genes. And when we look for the gene pathways that are enriched with these lead sites, we see here developmental processes are enriched as well as nervous system processes. So even in the mothers, we're seeing gene targets that are related to nervous system or calcium signaling, which is important in the nervous system. We understand that these findings we need to replicate in order to be confident in the rigor of our results here. We were not able to identify another study conducted in pregnant women for these metals. So we were looking at comparing our observations in women's blood to some other tissues observed by some of our colleagues and discussed here, including placenta, led by Todd Everson in one of Carmen Marcet's studies, Xiaowei Wu, I think that was Project Viva, and Andreas Cardenas' study with mercury, and those last two were in umbilical cord blood. So these are different tissues and different developmental time points. But in general, when we restricted to, particularly when we restricted to sites more highly associated in our study, we observed that there was some concordance here. So we need to do further replications testing, and to that end, there's an effort being led in the Pregnancy and Childhood Epigenetics PACE Consortia by Mariana Bustamante from the INMA cohort, which seeks to look at mercury and methylation, in cohorts across the globe. So I think we're moving forward with efforts to test these associations across multiple populations, and this will be exciting as it goes forward. The second study I'm going to share with you today is about as a toxicologic approach, because we wanted to dig a little bit deeper. Because one thing that it can be challenging in an epidemiologic framework, we often will measure just a mean level of methylation, so an average across all the cells in the sample. That's not necessarily reflecting the levels present in any cell. So a cell might have very different levels. Here, each dot would be a cell, and the average would be what we're measuring in a methylation study. If we're observing an association with metals exposure, here, for example, an increase in methylation with an exposure in this hypothetical scenario, we might have a situation where there's a direct effect where that metals exposure is increasing methylation across all cell types. We might have a scenario where there's just one vulnerable cell type that's driving this change. Or really, we might have a situation where there's no effect on the epigenome, but it looks that way because we're observing a change in our cell type heterogeneity. And all of these are very important biologic effects of metals on the system, but they are working in different mechanisms or different pathways. And so we want to look to this more closely. To investigate this, I collaborated with Dana Dolnoy and Justin Colosino as part of the Target 2 consortium, which is looking at perinatal exposures to many compounds, including, in this case, lead. So in this case, during the prenatal period and early weaning period, the mice were exposed to lead, and they were aged up to 5 months in the offspring. We were able to scavenge as part of Target, they're looking at many tissues, but we were able to obtain the hippocampus from these mouse tissues and dissociate them to a single cell suspension. And this is an important step because this allows us to look at cells individually. And we use a platform called the Chromium 10X genomics system. And we were feeding these individual hippocampus cells into their microfluidics device, where each cell is able to be treated individually, and the RNA transcripts in that cell have a barcode attached, so that when we conduct RNA sequencing, we're able to recapitulate and identify which cell those came from. So in this way, we can identify whether the effects of lead are happening either across all cells or maybe in certain vulnerable cells or maybe in certain cell types. And here's what the output from some of that data look like. You can think of this kind of like a principal components plot, where each point is a different cell, and the points or the cells that are closer together have more similar patterns, and those that are farther apart are more disparate. And you can do clustering algorithms to kind of categorize which cells are more similar. And then you can take on what you know about key characteristics of the cells in that tissue and map which cells there are. So here we're mapping one cell type, microglia. Based on the known expression patterns, we could assign cluster membership to oligodendrocytes, endothelial cells, neuronal cells, et cetera. And we can do this cell by cell. And we can estimate what proportion of cells we're able to capture in our control mice or the non-lead treated mice. The next step, we take that exact same plot, and instead of painting the cells by which cell cluster they're part of, we paint them by whether they came from a lead or a control mouse. So in this case, blue is lead, red is control. And the first thing I notice is that there's an uneven spread across all the cells. So there appears to be clusters by the different colors. And that's what we find when we quantify it. So we observe that in lead-treated mice, there's an enrichment, or there's an excess of oligodendrocytes observed relative to controls. And this really turned me on my head. I came in expecting to observe effects predominantly in neuronal cells, and I was surprised to see this effect where in perinatal period is associated with these persistent changes in the glial cells. And you can also take this data and collapse across all cells and do differential gene expression testing, kind of similar to what we would do in a bulk tissue analysis. And here we observe that those lead-related gene expression targets were enriched in psychiatric disorder genes, which is provocative in terms of thinking about the types of genes that are have this latent change in expression. You also, with this technology, we can look at cluster-specific changes. So we can look to see what is actually happening in the transcriptomes in these different cell targets. So today I got to talk to you about two stories that I'm really excited about. The first was an epidemiologic framework looking in pregnant mothers with blood levels of manganese and lead, and we observed also in the blood, we observed differential DNA methylation that mapped to several key biologic pathways, including immune signaling, metabolic processes, cellular processes, and neurodevelopment, which have implications for the pregnant mother. And we also did a toxicologic study looking with perinatal exposure in mice aged out to 5 months of age, and we observed persistent changes in cell proportion and gene expression, particularly among the glial cells. And I think being able to do studies of lead and molecular effects jointly across both disciplines of epidemiology and toxicology really enriches and informs our work. So thank you for your attention today, and I'm excited to have had the chance to talk with you all.
Kelly, thank you so much. It gives me so much pleasure to see you give this amazing talk after also knowing each other back at Hopkins and realizing we've had like 150, 160 people here, and so many of us are connected. If we develop a network of our connections, it would look amazing. And I think it was really beautiful how you showed how important it is for us to work together in epidemiology and toxicology, because a lot of the limitations that we've discussed with causality, with interpreting the signals, really the experimental models are going to be so necessary. So I think that's really great. And I see there are some questions, so I have my own questions myself, but let's go through the questions first. I think it will be great. So Carmen has a question saying, wondering if you have or could look at lead uptake in some of these specific cells. Some epidemiological studies suggestion lead accumulates in brain and might be interesting if that explains oligodendrocytes.
That is a great suggestion. Thank you for that, Carmen. We used every last ounce of tissue we could. If I were to go back in time, I would not do my first single cell RNA sequencing experience on such a tiny tissue. So unfortunately, we used the entire, both hemispheres, and we've proposed and have in review with NIEHS a proposal that would allow us to redo the study and also fix half the tissue so that we could look and confirm histologically, are these changing, these cell distributions happening? And I think your suggestion to look at the deposition and the localization of lead relative to cell types. I've seen other papers in human brains where they've shown that their lead, these were particularly multi-metal studies, but they've shown that metals are non-uniformly distributed in the brain. There's tiny pockets and clusters, and like there's pockets of iron in the brain. And I'd be interested to see where lead would go also, and where that would be relative to other cell types. So thank you for that outstanding suggestion.
Very good. So there are a couple of more questions. One from Corinne Mann. She said, do you think, amazing single cell data, do you think it is a question of real cytotoxicity or interrupted differentiation, pseudotype?
Yeah, I've been trying to grapple with this. Do we think that the oligodendrocytes are expanding, or do we think that the other cells are undergoing apoptosis or some kind of damage? And as part of this new study we're proposing, we'll have more time periods. So this is kind of just a snapshot of what's happening at 5 months, but I think we need to know earlier on in development what's happening with those other cell types. We did have, we were able to look in a, publicly available data in a neuroprogenital cell culture model. And we reanalyzed their data where they did 3 levels of lead over, they did RNA sequencing every day for 29 days. And they saw an expansion of a lot of these oligodendrocyte marker genes, similar to our data. So this is in a different species, it's human neural progenitor cells, and it's a different model system. So I think there might be something to this. They did not know apoptosis in this model. I think the lead levels were not large enough to induce that. So this is suggesting expansion, but we would want to look also in the mice and see if we can observe that phenotype.
Very good. So the question from Kelly Tamashiro, intriguing data, were the mice female or male? Did the offspring have cognitive deficits in adulthood?
Oh, great question. Yeah, so this was a relatively small study. In each treatment group, lead and control, we had 2 males and 2 females. So we didn't have a sample size to be able to do sex stratified analysis. That would be a crucial area. We know that lead has sexually dimorphic effects. We know that risk for many of these psychiatric disorders is sexually dimorphic. So that's a really important area to go in. And Dana Dolinoy's group, not in this mouse cohort, but in other mice using the same treatment paradigm, she has observed sex differences in several behavioral characteristics, including feeding behavior, overall activity levels, even in mice which are not experiencing weight gain. So there's there's a basis for behavioral differences in this model system. We have not in our group looked for cognitive differences in mice, although that has been previously documented with lead exposure during the perinatal period in other groups.
So Brandon Pearson has a question from fantastic work. Can you be certain that your cell markers from the RNA-seq data are not under regulation within non-target cells? In other words, is OLIG1 upregulated in neurons by lead? You would falsely conclude they are more oligon when in fact many cell types have changed expression of so-called marker transcripts.
Yes, thank you. And I think we are relatively in early days with our single cell analyses. We used outside data from the mouse Allen Brain Atlas to identify marker genes for all of these cell types. So this were an outside resource that then we were applying to our data so that we weren't generating our cell type specific signature within the same data set. We're also looking to do spatial transcriptomics in the future, which would retain the spatial architecture of the tissue, and you could actually understand we have our all-in-one gene expression. It's happening in an actual oligodendrocyte cell. So that's a technological advancement that's become available since we first did this study. So no, I think I'm not 100% confident, and I want to be validating this in multiple data sets and also be validating it in situ in the actual tissue before. But we have a building database of marker genes for several of these cell types, and if you look at it, they're quite distinct. It's like 5,000x difference in expression for those cell types based on those reference data. The other side thing that I would say the single cell approach really helped us is we were able to identify one of the 8 samples had a little bit of choroid plexus cells sequence in addition to the hippocampus. So you can pick out if you have some non-specific necropsy or tissue identification, and you can remove those cells post hoc. So we could identify, oh, these are those cells, that wasn't our target sample. But if we were to do a lead versus control test on that, we would have all these cell type specific marker genes coming out. So by doing this, we can refine our analyses and make sure we have a more clear tissue specific approach.
Very good. So we have 2 more comments. One from Sasha Nicholson. She said, I've heard that astrocytes serve as a sink for lead. That's good.
Thank you.
The second comment is from Shere Farzan. Have you looked at whether the metals-related methylation changes you see in pregnancy are associated with any pregnancy complications, for instance, blood pressure or gestational diabetes?
Oh, that's a great idea. I tested the lead response genes for association with psychiatric disorders because my primary interest is the brain, but we should do a wider screen for pregnancy complications. Thank you.
Very good. Okay, wow, lots of questions. I had myself a question that the fact that you had more sickness with lead and mercury, do you think that's generalizable? That's specific to your population? Do you see the same in the animal models? What do we know about that?
We saw more signals with lead and manganese, and I was unable to find another paper that had done a manganese EWAS, but consistent with other lead EWAS, there was a strong signal there. I've heard in the published literature as well as anecdotes from other folks who have this ongoing that lead does seem to have a nice robust signal, and indeed we've seen this in the talk earlier today, which was exciting, that the lead signal was strong enough to be able to be used for classification of a biomarker signal. I'm not sure, and what I can say is that these exposure levels are in line with what the NHANES levels were, but we could imagine globally have very different exposure levels. And indeed, that's what the folks at PACE who are trying to do the meta-analysis of mercury right now are observing, that it's particularly challenging to conduct meta-analyses with environmental exposures when all of our individual cohorts have almost non-overlapping exposure levels. So trying to think, do we need to do some kind of weighting? How do we evaluate the heterogeneity and signals that may be coming from multiple cohorts?
Wonderful. So thank you, Kelly, so much. And something I want to announce to everybody in the audience is that unfortunately Alison Kopsko couldn't join us today for some unexpected reason. And so that's it for today. We thought that we would not, and we just learned it just a couple of days ago, and we thought we would not announce it because we thought people would need a little bit more time anyway, and we might have been late. But it seems that we are doing pretty well on time. And actually Alison would have had perfect time to go over her presentation and then for us to have the overall discussion as we wanted. I think Kelly mentioned earlier that this was a need. And I think Andres and I were very selfish. We did this together because we thought about this because we needed it ourselves. We have so many questions, so many issues, so many different metals and tissues and ways of looking at epigenetics, that we wanted to see kind of this global, of starting to have a global picture of what's going on, what types of data do we have, and start the conversations to see how can we move this forward, how can we be more connected, how can we work better. And I think that's what we are here now. And Andres, if you want to start the conversation, I wanted to call on Andrea, because, you know, as his name has come up several times, he's a real leader in the field. In here, he has put us together in many ways. And but unfortunately, I don't know if he's back, because he had to leave just a few minutes ago to have a meeting that he had scheduled. But if he comes back, it will be great to hear from him. But Andres, if you want to start maybe framing the conversation and some of the things you've been thinking about, and then we can hear from others. Whoever would like to talk, you can raise your hand or just chat or just interrupt, unmute yourself, be brave. Everybody, we still have 133 people here. Don't be shy. All opinions, all voices are important.
Thank you, Anna. I just want to follow up with that, you know, and thanks to speakers. I think we had a lot of variety and really, really great work. So thank you to everyone for participating today. I think Kelly kind of provided a really nice ending talking about the meta-analysis that is planned in PACE with Mercury. And some of you participate in the PACE consortium that's mostly around prenatal exposures, although they haven't done much on exposures besides smoking, smoking biomarkers. So, I mean, some of the things that Anna and I are working on is trying to integrate epigenetic data and kind of moving towards this meta-analysis of epigenetic data. And I just like to take a brief survey of some of the participants to see if people will be interested in doing that. We're kind of trying to do this with arsenic right now. You know, Elena's expertise on biomarker development. I'm also curious to see her opinions and others' opinions about whether we can do this in a consortium type of way. You know, and also with Carmen and others about, you know, the novel biomarkers. I know a lot of people are doing microRNAs as well, and some people have placenta data as well on top of cord blood that is a little bit more novel. So I just want to invite everyone and also kind of get a feel of how people feel about participating in meta-analysis. I know it's a lot of work. A lot of times this is unfunded. So we try to really, you know, do what we can in some of this work. So for now, I would like to open it up to the, you know, the speakers as well as everyone else in the call. As Anna mentioned, feel free to unmute yourself or write in the chat. We really want to make this useful to you and to the field, you know, so whatever, all ideas are welcome. We'll try to take notes and really try to do something about it as well. So I don't know if someone wants to go first to refer to either meta-analysis efforts or other ways to integrate data.
Andres, this is Carmen. The one thing I was thinking of as we were all talking was that you know, a lot of this is still single metal analyses too, and I think we might be missing a lot by not starting to think about how mixtures of these metals are important. You know, and we've only done a little bit of that, and I you know, I think we still don't know exactly the right ways to do everything around that. But I think it would be really interesting to start to explore that. I think we have this data, and that might be sort of an approach that we can think about, you know, as meta-analyses, right? So instead of each of us trying to develop our own way to do their mixture and then to look at their mixture and to maybe think about, you know, that would be very impactful, I think, if we could come together to do something around metal mixtures and then to think about that related to, you know, these epigenetic changes, whether it's cord blood methylation or you know, whatever we want to look at it. You know, that might be something that where we can really make a bigger impact on understanding of what's going on. And then we could make Kelly have to figure out how to do that in an animal, so she could.
I feel like we put always Kelly up for all the challenges that we get. No. Sorry, Kelly. Yeah, and I think, you know, Bob's lab has been measuring a lot of metals, and we also have multiple metals in Project Viva, and I know the Protect cohort also has multiple metals. So they're part of the SRP as well. So you know, I don't know whether you know, there's methods and some of the methodological people like Arce and Elena, you know, whether we can really do this, integrate the multiple metals as Carmen was mentioning with epigenetics. I know this is a very high dimensional problem because we have the high dimensionality of the metals and the high dimensionality of the epigenetic data. So I don't know if you have any input on ways to do this, Elena or Arce.
Thank you so much for this question, because actually I'm working right now on mixture approach for expanding to EWOS. I never done it on a methylation CPG sites, but I'm working on that. There are very good methods outside. I mean, we all know BKMR, WQS, and also other methods that are more novel, like the quantile-based computation. However, those are extremely time-consuming, and it might not be possible to apply those methods for the entire array. So we are trying to come up with a more efficient way, more computation, less computationally intensive, but we are still working on that. So I don't have an answer for that right now. Sorry about that.
Just to add to Carmen's comments, sorry, this is Corin. I think one of the other things that you might consider as you're going through this is the interplay between essential and non-essential metals. So add another layer onto your mixtures is to make it more complicated for those of you that do this, is whether or not you're actually having this interaction between a metal that we would actually see the toxicity because you're impairing an essential component.
Hello, this is Kiran. I was also wondering if these big human studies that have been carried out in the past, if like along the same lines as Carmen, if they have collected data about other metals from these populations, and be nice to like look at those data first before actually, you know, designing bigger studies for combination metal exposures.
There are many studies that have multiple metals. And as we are learning now, some of them have epigenetics, some of them don't. They might have in the future. What I am thinking as I am listening to everybody and the conversation, I think it will be very useful to have like kind of like a library or a map of the different data that are available. Which studies, if we just think of metal epigenetics, because things can get really difficult very soon, and this is already difficult enough. But if we just think of metal epigenetics, which human studies and animal studies, I think it will be very useful to have those side by side. And do we have which studies are out there, which metals were they measured, in which biospecimens were the metals measured, which health outcomes, which types of epigenetic data in which tissues, blood, placenta, cord blood, cervical cells. It seems incredible amounts of different tissues that are available. So, but as we have those mapped out, then I think it might be extremely useful for people to know, can I replicate my findings? Can we do a meta-analysis for a particular question, metal mixtures and a particular health outcome, with blood DNA methylation or with something else? So I think that could be an amazing resource. And I wanted to bring and thank actually the NIHS Superfund Research Program, because it's thanks to them that actually we are doing this today through their data external use supplement that they had in the last year. That's what allowed Andres, myself, Mary Gamble, and Bozak, and others to start having conversations through that relatively small project. And then we thought, oh, let's have a workshop. And we were going to do it in person at Columbia or somewhere else, maybe at the time of a conference. And then of course, our world changed completely. And we thought, what do? Do we wait 1 year to do this? And we said, okay, let's try to do something remotely. And I think it's been even better because honestly, how many people could come to New York to spend a day or so? Much harder. So I think I am really happy that so many people are here. So I think if we start thinking how to map things, know where they are, then moving forward with meta-analysis, pooled effects, replication across studies, integrating between toxicological and human. So I see really a great potential here. And I think we need to think of which sources of funding can support some of these efforts, because it's almost impossible to do this without at least some funding. But I think there might be opportunities there. And I think Scott Burkiel has some ideas.
Thank you, Anna. I'm learning a lot. Thank you so much for this, Andres and Anna and others. This has been fabulous because, as toxicologists / immunologists, much like Corin, I have to agree. I saw a paper presented at the ICT meeting in Hawaii last summer when we could still fly. And it was a very interesting work done in Mexico City with looking at methylation, both of some of the line 1, but also some target genes, looking at global methylation. But it showed how some of the metals that Corin was talking about, this is Betsabet Quintanilla's paper, it's published. But she showed that the metals do modulate each other, the so-called healthy metals versus toxic metals, which I'm not sure I like the term, but she's looking at selenium and zinc and some others and showing how they really do dramatically affect whether you're looking at cadmium or arsenic or what you're looking at. She found major effects. And so I think it's complicated, right? It's muy complicado. But the upshot of all this is that I appreciate that approach as we're doing this, starting to do this in our group, look at metal mixtures and effects on the immune system. It becomes something else I learned today, because a lot of people are just taking blood cells and doing methylation on blood, PBMCs and the like. But how important it is to do the target cells. And when Andrea Baccarelli and I have talked about doing some of these studies, we're thinking we have to actually, sort the cells. A B cell is not the same as a T cell or certain type of a T cell. And you guys are starting doing work on, brain regions, which is super difficult. The single cell, I think people got very excited about the single cell data you showed today, because, and Kelly, I like the way you explain both toxicology and epidemiology, because I've been trying to learn this new world, and it's tough. Even I have good teachers like Anna and others that it's tough. So the upshot is that I do feel like we need to keep talking as a group and doing this. And when we eventually do have face-to-face or have a day-long or more meeting, I know SOT is looking for a program now, not knowing what's going to happen next spring, right? But there's Andrea. So I would just say thank you to everybody. I think it was a learning lesson for me. The technology is, changing, but you all are bringing, and the mathematical modeling, some of the pathway analysis, like you were doing, Arcy, with the cadmium on your EHP paper, which was beautiful. It's just, it really speaks to me, because I don't get all these ways, even the stuff that was being shown by Molly Kyle with all the different approaches to modeling. I mean, it gets up here very quickly for me. But I think these are powerful approaches. The data has to be robust, and I appreciated whoever it was that talked about reproducibility. I really appreciate that, because there is this concern that, these are just one-off observations and that no one could ever reproduce them. So the more we lock in some of our understanding about the approach, and knowing the exposures is key, and again, looking at metabolites is so key, because they mediate so many of the effects. So I've said enough, but thank you very much for educating me today.
So, Anna, this is Bob. I just wanted to make a couple comments about metal mixtures, because that's what I've been doing for a long time. I think we need to make some distinctions between replication and reproducibility. They're not the same. And I have a lot of concerns in the epi world that replication may, because of genomics, be a gold standard. And I don't think that's a reasonable gold standard, and I'll explain why. you know, one, you have to measure it at the same life stage. And a lot of studies, you know, unless they're planned, they're not going to measure the same elements at the same life stage. Ideally, you should be measuring it in the same biomatrix. If you measure it in blood versus urine, they're not necessarily combinable. And third, my study is in Mexico City, and the covariance structure of the metals there is probably very different than a study in New York City versus a study in Rhode Island versus a study in Europe or Asia. And so you might even predict that you wouldn't replicate the finding because the metal mixture that really drove the effect in Mexico may not even be present in Italy. And so I think what it really says is we need collaboration with toxicologists, because I think replication and reproducibility and biological plausibility will almost have to be done, at least initially, with in vitro assays and maybe ultimately with animal studies. But I think in vitro probably is a better place to start because of all the possible combinations. And then toxicologists need the epi data to know what are the relevant mixtures, because you know, you don't want to study something that doesn't happen in real life. And you want to study it in the right proportions and at the right life stage. So I really think this is a question that really speaks towards program project grants and integration and collaboration across different types of studies. I don't think you can actually get to the truth unless you do that.
I totally agree with you, Bob, and I think by comparing across human studies, you can see which signals are consistent and which ones are not. And maybe look a little bit about the sources of heterogeneity. And maybe we can learn something if we can identify some of those sources of heterogeneity. But looking together side by side with the toxicology, I think we are in this together, and we can see this from the audience here, that there are lots of toxicologists today, and maybe Andres and I were a little bit biased today because we have more epidemiologists and toxicologists. So we are very happy that Kelly brought this final presentation, which was really, I think, very illustrative, showing how powerful it is to have both side by side. So I totally agree on that. Thank you, Bob. And Andrea, we've mentioned your name earlier, and we've mentioned that, you know, you've been instrumental to many of us. And so maybe if you want to, and we've been talking about how can we work better together, keep this going for the future, maybe, you know, meeting again from time to time, remotely, inviting other people to be able to present. We had a small set of presenters. I am sure that in the audience today, there are amazing people who could have lots of really amazing things to share. And so we, you know, if you want to say a few words, Andrea, your experience, how can we have full efforts and more efforts together?
Yeah, no, I mean, first of all, it was I missed I think the most of Kelly's presentation. Sorry, Kelly, for I had to step out for a meeting with the CTSA, but I'm sure it was great and amazing. But then a couple of things. First, this was incredible. I mean, it's to be honest, I think sometimes we travel thousands of miles to go to a conference, and we don't get anything close to this quality of presentations and discussions. So really great ideas. A couple of things. I would love to do it again. I mean, if you guys do it again, I would love to be there. And also something I would like to mention that I got inspired. We were inspired by your leadership on this, and we put together a group that in August is going to meet to discuss multi-omics. So there are people that I know are interested in multi-omics from all over the country, and including many who presented today or are in this call. So if anyone of you is interested to, and mostly the idea is now we have not just DNA methylation or transcriptomics, but we start to have many different data, genomics, DNA methylation, etc. How do we go about analyzing the data? So it will be mostly, I think, presentation and discussions to share ideas on how to tackle the data. So I want to say because it came to my mind while you were speaking, and if anyone is interested, please let me know and send me an email that I'll be happy to loop you in. We have a date that I can't remember at the moment, but it's going to be 3 hours, I believe, also in the morning in August.
So what we can do, Andrea, is because Nancy, who truly has been instrumental in making this possible, she has the list of everybody who has signed in for today's symposium.
Mm-hmm.
So we can send them information.
Absolutely.
Once we remember the date, so that people know. And then we can at least create a list served with everybody who was here today, so that when we have new connected, of course, being very mindful of not bombarding anybody's emails, but just when there is something really relevant as what we are doing, so that people can be aware of this activity.
Anna, something else that I wanted to share was the Human Health and Exposure Analysis resource here. It used to be called CHEER, but this is how I've been able to work with Bob, looking at several metals. So other cohorts that perhaps have multi-omics tools and they're really interested in adding an exposure component, I shared the link on the chat as well. The other thing that you mentioned about knowing who has what and what they're doing, this is, the symposium is helpful for this, but it would be nice to have like a list, as you mentioned. I don't know if Elena or, Bob might be willing to share some of their experiences with CHEER. I know, Elena, you're heavily involved with the data core. But this is really nice. I mean, they have a portal where you're able to see the studies, the sample sizes, the number of analytes that have been done. I mean, it's done extremely well, more so than what we could do right here, starting like a Word document or something online. But Elena, where do you see is the future of that? The data will be publicly available. This is my understanding, right?
Yes, correct. It's actually a nice opportunity to have environmental data, to add environmental data on your data set. I'm involved in the data center, so I will perform statistical analysis on some of the data. And data will be publicly available after 1 year, 2 years from the latest publication, or I don't recall exactly, but the data will be publicly available for everyone. So we can move, the idea behind is that we can move science forward using publicly available resources, including data. So people can create more methods and also to combine multiple studies and understand better the role of environment on the set of diseases or long-term health effects. So this can be extremely helpful.
That sounds like a great model. One concern that I have regarding the data component is that many epidemiological studies cannot really share the data. For instance, the Stronghold study could not do that. And so there are others that cannot either. However, there are ways of accessing the data. It's just that we cannot post it there. So at least if the information is included in that portal, in that web portal of how to access the data, that's still very, very useful for the investigators. I noticed that, so
Anna, can I do a brief public service announcement on here, CHEER? So CHEER has changed its name to HERE, so instead of the Child Health Exposure Analysis Research, it's now the Human Health Exposure Analysis Research, which means in the past we only accepted studies about children's health, now we accept studies across the lifespan. And the way it works is it has to be either a clinical or epidemiological study involving human samples. And if you have received NIH funding, and that can be very loosely defined, but if any part of the study was NIH funded by NIHS specifically, you're definitely in. Some of the other institutes like NHLBI and NCI, and if you're in the ECHO program, those are also supported. You do a relatively brief application. You tell us what samples you're interested in submitting and what you want analyzed. There's a consultation phase where we evaluate whether the samples are useful for what you propose, but also sometimes we say, maybe you could try this instead. So that there's some really nice back and forth and collaboration that develops out of that. And the samples are analyzed free of charge to the investigator. So that's another advantage of it. You don't have, it is basically a grant. You can't take salary support off it, but you will get all these samples for free, and you'll get support from Elena and other statisticians on analyzing that data.
Wonderful, and people should really take advantage of that. Something I was going to briefly mention is that people have been posting great comments and links in papers. What I am thinking is I hope we can save the chat, and so that we can put together that as a kind of a summary of different links that might be useful for people. So, because otherwise when we leave, the chat will be gone. And I think it will be nice to just keep this chat for the future.
I want to take pictures because I understand that our IT decided we cannot save the chat.
Oh,
it's
wow.
A huge security threat, I'm sure.
But it's such a long chat that taking pictures might be difficult. Okay, we'll see what we can do.
I just tried to save one yesterday and I failed. The function has been locked by our IT.
Yeah, because before that was not the case. Why did they do that? That doesn't make any sense.
Security always finds new needs.
Okay.
I wanted to also briefly ask Maria and Arce as well, if you see if it will be useful to have type of a meta-analysis of arsenic exposure and DNA methylation, whether some of the cohorts that you have been working with will be willing to participate, as well as Carmen perhaps, and whether Maria might have more expertise on whether we need more of a prenatal EWAS meta-analysis or on an adult EWAS meta-analysis, or whether we should do both. How should we approach this issue?
Yeah, I think that's a great point. With respect to, the predominant literature right now, I think you've highlighted the two main areas where we've seen the most publications. Quite a bit of work has been done looking at prenatal exposure with umbilical cord DNA methylation. And again, I think a meta-analysis of those studies would be tremendously useful since many of the early studies were pretty small in size. So there would be, I think, a tremendous strength in pulling the data across those studies, and then also to begin to look at some of the sex-specific effects. Because I think none of the early studies really had an opportunity to do that individually. And then again, looking at concurrent exposure in adulthood with DNA methylation, I think would be interesting. I think the challenge there is going to be there's really a lot of variability in terms of exposure levels and the exposure biomarkers used. So that potentially would require, I think, a bit more thought in terms of how we can combine data across the existing cohorts. But I think looking at prenatal exposure would be a great place to start for arsenic.
And I think going back to the idea of the mixtures versus the single metal, I think the mixtures is amazing and we have to do it. And there are new methods, as Howie was mentioning a couple of methods, and lots of great expertise. At the same time, we need to recognize that the mixtures are even more complicated to understand the sources of heterogeneity because of how different the mixtures are and across different populations. So one smaller advantage to start some efforts with single metals like arsenic or lead is that at least it's easier to start thinking which are the relevant sources of heterogeneity there. And so, I've done a lot of single metals in my life, and I know how difficult those studies have been and how many issues and problems there are. So I have a lot of respect to doing things simple, because simple becomes complicated very fast. And so, I see value in, of course, in both approaches, and the mixtures are super important. But maybe starting with arsenic, lead, and bringing in people who are really interested, motivated, who can think hard of how to solve the complexities, I think could be good. And of course, the mixtures too. I am not saying that the mixtures are not good. It's just recognizing the challenges. Terence has a comment.
Yes, thank you, all of you, for really wonderful presentations. And as Scott said, I also learned quite a bit. It was really great. I'm wondering if we can explore one technology that might be able to answer some of these questions about single cell analysis, mixtures, and relevant expression biomarkers, both at the protein and maybe even at the mRNA level, depending on sensitivity. And that would be laser ablation ICP-MS. I know Andrea and Bob, you guys have done that at Columbia for a number of different, and at Mount Sinai for a number of different projects. And it seems to me that that technology is ripe for exploring to answer some of these questions.
Maybe some of the people who have used it would like to share some examples or some experiences.
Well, Manish Arora developed the technology. Manish is a dentist and a chemist, and basically it turns out teeth grow like trees, and there are growth rings that are formed on a daily basis, like the annual growth rings that a tree forms. And you can actually histologically identify the ring that was formed on the day you were born, and then once you orient yourself in time, if you sample in one direction, you're sampling prenatally, if you sample another direction, you're sampling postnatally. So it does afford a way to measure metals in a very integrated fashion. So, you know, the other problem with, you know, any environmental health study, you know, we measure, you know, blood phthalates or urine phthalates or blood lead one day in the third trimester, and we think that day represents all the third trimester, or at least we assume it does, but I'm sure we all know it doesn't. And, you know, there's some error introduced because of that. With this method, you can comprehensively measure all of the third trimester, and you can average what it is, or you can even look at specific weeks and what it is. So it does allow a degree of temporal variability that isn't achievable in any other way, because I don't think any person in their right mind would come in every 3 to 4 days to get a blood or urine sample drawn in pregnancy or postpartum. So in that way, I think it does provide a lot more information because that temporal variability is reduced. And in fact, in most of the studies we've done, we're finding statistically significant results with sample sizes less than 100. And I believe that's in part because the exposure misclassification is greatly reduced in using the tooth biomarker, and therefore you have more power, because that error that we get, which is just, you know, part of being able to do epi studies, as I said, nobody in the right mind is going to come in every day, it gets reduced. And I think that that is actually a big advantage. Now, it only works right now with elements, but Manish is developing methods to look at organic chemicals, but that will have to be validated to make sure that it really does represent what we think it does with respect to the dates. But that's something that he's currently working on.
So if I could just add one thing there, I know that Gary Nolan and his group have worked on doing metal tagged antibodies where they use the stable isotope lanthanide series metals. And that together with analysis of the environmental metals might be very useful.
Yeah, I think this is slightly different than what you mentioned, but Manish also has a method where he tags antibodies with elements and uses the ICP-MS actually, and the antibodies are against specific proteins, but he can look at the two-dimensional distribution of the proteins, but also he can create two-dimensional maps of elements in any tissue. And it's quite possible that it's not just the concentration of a metal or an element that actually predicts health, it's the way it's distributed. And two people with the exact same concentration, the distribution might tell you more about who's at risk for disease than the actual level, which is also something that, you know, it's very hard for us to do. But I think by working with toxicologists, I think those are questions we can actually start to address.
Thanks.
Excellent. So we have 6 more minutes officially before we finish. So I want to make sure that if anybody who's there has something to share, something you would like to say, please feel free to do it. Don't be shy.
Hey, Anna and Andreas, can you hear me okay?
Yes.
This is Michelle Heacock, not being shy. Just to thank you both for putting together this great group of speakers. It was so nice just to kind of get a really wide swath of the kind of research that's out there, but also very scary to know that there is a lot of things to consider. As you mentioned earlier, you know, one of the things that was the driver for this particular workshop was your activity where you and Andreas are working hard to try to bring together some data sets from separate cohort studies and learning, and through that, how can you best, you know, come up with recommendations so that people are better poised to be able to do that in the future. I mean, we certainly understand that there are significant barriers because of personal identifier, you know, PII. We don't want to, any way, you know, compromise that. But I wonder if you could speak a little bit about, you know, how you're tackling that and sort of some of the recommendations that might be percolating up, and maybe if some of the people on this workshop webinar might be interested in making some contributions.
Thank you, Michelle. Thank you so much. And thank you again for the Superfund Research Program for initiating in a way this effort. And I think the problem with the personal identifiers is that it's not, at least for some of the cohorts that we work with, studies that have tribal communities, indigenous communities, it's not even enough to remove the personal identifiers. Even with removing them, they don't agree with the unrestricted data sharing, and they told us over and over and over again. I know that applies also to Scott Burkiel and the work in New Mexico, other cohorts. I can imagine even non-tribal communities might also be very worried about that. I think they have the concern that the data might be used in the future in ways that we cannot anticipate today. The information that we have on the epigenetics, the genetics, there is a lot of hidden information there right now that we don't understand. We are developing new methods and new tools that it's allowing us to understand those data better. We are going to be able to reanalyze this data in the future as we understand what the data means more and more. If there is something in the data that we cannot anticipate today that could be harmful to the communities in the future, they are very worried about that. And we as investigators have to respect that, because actually they could be right about it. And that's why we have to be very careful. So I don't anticipate that we'll be able to share the data through unrestricted platforms. However, we can have data dictionaries. The metadata is fine. They're very happy with sharing that, saying what's available. But the individual participant level data right now for certain population groups is not, we cannot share that. And I think Scott wants to say something also.
You're right, Anna. The Native Americans that we work with in Four Corners and elsewhere, that it's quite, it's virtually impossible. I mean, genetics is the key thing that they're worried about, right? And, but we're doing a lot more than genetics with the population, but even that data is hard to share. It's very, and it should be protected for many reasons. But there's quite a big difference between different, as you build trust working with groups and you do put in protections, there may be a possibility, but I can say it's quite different between different populations. Don't put everybody in the same, you know, cup, basically, about what the reaction should be. So just to say, yes, it's very difficult to share any of that kind of data, but you can go through the laborious process of getting approvals for a specific data set, and people are convinced that it's safe, that they're protected. But I just think to have any kind of global things, just share with everybody, would be very, very difficult.
Exactly. They are very happy to share. And they are always say, don't get us wrong. We are very happy to share, but we need to know how is the sharing happening. So that's why the unrestricted platforms do not work for them.
And I just wanted to add to Michelle's comment. I mean, with this, through the Superfund, we have been working together to join the data, meaning that we're kind of conducting analysis separately and then coming together, but really trying to standardize pipelines and methods and approaches. So that's not entirely data sharing. I mean, we are each approved by IRB to do this locally, our own institutions, but I think this is a way forward, at least for some of the genomic data. For the exposure data, that's a little bit more unclear, but some of you might be familiar. Perhaps it was a recent paper, I believe it was in Nature Communications, looking at identifiability of epidemiological data. So even if you share epidemiological data, that can be challenging. But so far, the Superfund has really allowed us to collaborate, kind of like on a weekly basis. So persistence is important. Talented people are also very important. So we have Anne Bosak helping us, as well as our trainees here. And just having this motivation to keep moving forward with integration too.
And Andres, I think those are beautiful words and a wonderful way to end this event. Thank you so much for joining. This was fantastic, and to be continued. We'll be reaching out to all of you.
Be in touch.
Thank you. Bye-bye.
Thank you.